right. So I believe what we're going to do next, right, as we return from lunch. Before lunch, we had started processing a new data set, and we had also been discussing Ab-initio reconstruction 2D classification in the context of our ATPase molecule, sorry, not ATPase, AAA plus Unfoldase that was shown to be in 2 conformations. Before we left for lunch, we had started off in our Unfoldase projects. Everyone should have started off according to these instructions that we were following before. 2 refinements, one for the inactive symmetric 6-fold symmetric state of the structure, and one for the active state of the structure, which is asymmetric. And what we're going to do now is just briefly look at those 2 refinements and see what we got. So in one of them, in my case, for everyone who's following along in the practical, you would have done this in your own project, and for everyone at home, you can just watch what I'm doing. This refinement, we started from one of the classes of this 3-class Ab-initio reconstruction. That was the class that had the symmetric structure. And that refinement with symmetry enforced, if you recall, the parameter that we set was to enforce C6 symmetry. When we enforce symmetry in refinement, each of the particles is used multiple times during the reconstruction of the structure, and each particle counts for that many times as much signal content. And so C6 symmetry means that each particle counts 6 times, and we get 6 times as much information about the structure from the same particle data. So in that process, in our refinement that we did, if we look at the overview tab, we'll see the last iteration plots that we want to inspect in order to understand the quality of the structure, resolution, and its characteristics. So slices through the structure show that we definitely have some interesting details, some helical pitch is starting to be visible, and in Fourier space, everything looks as it should. This is the mask that had been used from dynamic masking during this refinement, and our final resolution structure that we got was about 4.2, 4.1 angstroms, which is great. With the symmetry enforced, that's a good resolution. And we can look at our orientation distribution plot, and we see that we have lots and lots of side views and a smattering of top and bottom views for the symmetric structure. In our posterior precision plot, which, as I mentioned before, is a different way of looking at the same information about viewing directions, we can see that every part of Fourier space is sampled at least something like 900 times, which is great. And means that we hope that the reconstruction is actually of a good quality. So to actually inspect it, we can go ahead and download the result right here. And we'll download the sharpened map since B factor sharpening was automatically applied. So I'll just download in a couple of seconds. In the meantime, we can look at the inactive structure, which was resolved, as we can see, to 4.5 angstroms, substantially lower resolution, but in this case, a fully asymmetric version of the protein that has that second molecule blurred out here and shows us the active version of the enzyme. In this case, we see the dynamic mask actually includes some of that floppy second structure, and we get to a final resolution of about 4.5 angstroms. Again, the viewing distribution, as we expected, has a wide band of views around the ring and then a smattering of top and bottom views, less than the other case. And now we see in this plot of distribution, posterior distribution, that here our distribution is a little bit worse because we don't have as many top or bottom views. We have not as many measurements as in the other case for all the directions. So we'll also download the sharpened map for the second case. And I'll open the first one so everyone can see what it looks like.
Just a point of clarification. Can you please clarify between the inactive and active states, which ones you're referring to? Like which one is which?
Sure, so the inactive state is the state where the molecule is by itself, a single molecule, and it's fully symmetric. That's the inactive state. The active state is the one where the molecule has a second molecule attached to it in the process of unfolding that second molecule, and it's asymmetric. Okay, so now we're looking at the reconstruction of our symmetric volume at a 4.1, 4.2 angstrom resolution. where we wouldn't expect to see side chain detail, but we would see helical pitch and the overall secondary structure of the protein. So with that symmetry enforced, we can clearly see all that here in our symmetric six-fold symmetric inactive version of the structure. At the same time, I can also open up the active version of the structure, and we can see the difference. Now we can see that in the active state, the symmetry is clearly broken. Some of the subunits have split apart in this ring to form a sort of like split washer type of configuration, broken the symmetry, and this cloud of dust is actually a second molecule that's totally blurred out because it's so flexible and floppily attached. But inside of this main structure, there is actually density, and according to the original publication with this data, I won't be able to find it right now, but there's actually density corresponding to the part of the protein that's being unfolded, the substrate. And you can see inside here, there is this sort of helical shape that's been taken on as the symmetry is broken. And that's part of this sort of unfolding mechanism to pull the thread of the second protein and unfold it. So that's exactly what we hope to achieve by separating these two conformations and refining them separately.
Ali, when you were examining these structures, did you download the map or the map_sharp out?
I always downloaded the map_sharp, and that's the version that corresponds to having the original reconstruction sharpened by the B factor that was estimated from this Guinea plot automatically. Like I said earlier, when you're doing this with your own structures, it's definitely a good idea to redo the sharpening with a different B factor, try a few B factors, and choose the one that creates the best visual quality. Okay, so that's everything we wanted to see in this data set. What I want to do next is just go through some of the advanced parameters of ab-initio reconstruction and explain what they do. So that's also complete. And for that process, I will clone this ab-initio reconstruction job and just talk through some of the parameters. So actually, before I do that, I will go back to the slides and talk a little bit about ab-initio reconstruction from a more theoretical mathematical perspective. We saw yesterday that what ab-initio reconstruction does is use a stochastic optimization algorithm. Oops. Sorry. Do you still see my screen?
Yes.
Yes, yes. Mm-hmm.
Strangely, my monitor has turned itself off. Sorry, just give me a moment. There we go. Okay. So yes, we saw yesterday that ab-initio reconstruction implements a stochastic optimization method. And that stochastic optimization method allows us to explore the space of structures to find the correct structure. And that's simple enough to understand in the homogeneous case, where we're just looking for a single 3D structure that best explains the data. In the heterogeneous case, however, there are a few extra considerations that go into the algorithm design and how it works. I won't get into too many details there, but the main conceptual notion is that when we're dealing with heterogeneity, not only are we trying to solve this sort of non-convex optimization problem where we're looking for correct structures in terms of the alignments of the particles and the overall density shape, we're also trying to explore the space of conformational variability. And those additional classes that we're looking to solve and that conformational heterogeneity adds a level of difficulty to the optimization problem, non-convexity, and it creates more local optima, more ways for the algorithm to resolve different results or get stuck in different local optima. You can think of the simplest case where you have 3 classes. One result may be to have 3 different conformations. An equally valid result would be to swap any 2 of them and create a different version of the result that is otherwise identical in terms of its significance, but mathematically different. And similarly, you could take any of the structures and rotate them, and that would create another valid version of the result, but a different version. Or if there are actually multiple clusters, you might find only 2 out of 5 or 3 out of 5. Or even if there are 5 clusters and you ask for 5 classes, the optimization may get stuck at a point where it has found 3 of those clusters and the other 2 the particles are absorbed into the first three classes and can no longer be separated. So there are many different ways for the algorithm to resolve different results. And a lot of that comes down conceptually to this concept of exploration versus exploitation. This is a common sort of theme in optimization methods for this kind of problem, especially for any kind of clustering or search or learning type of problem. There is a notion that when the algorithm is running and trying to find different conformations, at some point in its optimization, let's say it's found two different-ish structures that aren't perfectly resolved yet. They don't have high resolution details, and they're just at the beginning of optimization. It has a choice to either exploit what it has found so far and just hone in on those two classes and separate the particles on that difference, or further explore and try to see what other structures there may be similar to those two classes, or let them evolve further without getting too confident about the classification of particles. This is similar to what we were discussing previously with 2D classification, about how the level of confidence we have about classification determines what we end up finding in terms of the diversity of classes. And the same is true for ab-initio reconstruction. So in general, the algorithm will do something. We can somewhat modulate the exploration versus exploitation trade-off by using something that in CryoSPARC we call a class similarity score. And it is, in terms of the actual optimization method, there are a few like mathematical parameters that could be changed. We've kind of squashed those all into one parameter that has a somewhat interpretable value. So I'll just switch back over to CryoSPARC and show that. In our ab-initio reconstruction, we have the number of classes that we want to find. We can optionally choose to use a subset of the data at this point, not look at the entire data set. A minimum and maximum resolution we talked about yesterday. If you're working with very small particles or very symmetric particles, you probably want a higher maximum resolution value, like a smaller number, higher resolution. And the parameter that I'm looking for is called the class similarity score. This is actually not an advanced parameter, I believe. If you turn off the advanced parameter switch, you'll see it's still here, class similarity. And that parameter controls, this time, unlike 2D classification, this time it's a number between 0 and 1. 0 tells the algorithm that it is supposed to explore as much as possible. Try to find structures that are very different from each other, or in the worst case, if it can't do that, find one structure that grabs all the particles and the other classes with very few particles. So that's useful for looking for very different conformational changes, different molecules that may be present in the same sample, or totally different structures where we don't expect there to be any similarity between them. On the other hand, at a value of 1, this parameter tells the algorithm to exploit its knowledge at the beginning more rather than exploring, and try to find structures that are similar to each other but are exploring the conformational landscape of a single molecule. So let's say you're looking at, for example, in the case of the ATPase molecule that has a central stalk that rotates that I showed yesterday, we may want to increase this value so that we're forcing the algorithm to look for more similar structures that are more closely related to each other. At the end of the day, the algorithm is not forced to do that. It can still find very different things, but this parameter controls a sort of guidance that we're giving the algorithm in terms of what we're looking for. By default, it's set to 0.1, and that generally works quite well in most cases. But sometimes if you're looking for a specific kind of result or trying to tease out some other interesting kind of heterogeneity, you may want to play with this parameter. Coming back to other advanced parameters in ab-initio reconstruction, sorry. There are a lot of parameters here that are advanced, and most of them I wouldn't recommend playing with. I mean, you can try just for exploration's sake, but it's hard to provide an intuitive or explanation that actually captures something that's worth knowing. Symmetry can be enforced during a ab-initio reconstruction. As I mentioned yesterday, it's not recommended to do so. You should always try to do an ab-initio reconstruction in C1 first and ensure that the structure you get is actually symmetric, and then enforce that symmetry during refinement, not during ab-initio reconstruction. And yeah, that's the rest of the parameters are really very advanced parameters or things that really shouldn't be changed. So I won't explain any others, but there are a few more points that I want to cover in terms of what ab initio reconstruction does and how to interpret or think about its results. One very important concept that we should always keep in mind is that ab initio reconstruction cannot fix the problem of missing viewing directions, which we've discussed a few times now, the orientation distribution problem. If your data doesn't have enough or any views from certain directions, it is impossible for ab initio reconstruction to recover the correct result. That data, that information isn't in the data, and we don't have any prior information coming in from an input model or an initial model. So ab initio reconstruction can't solve the right structure. And you can always tell that this is the case by looking at the orientation distributions coming out of ab initio reconstruction. If they show all the particles pointing in the same direction, you know that this result cannot be trusted. It's only when ab initio reconstruction recovers a structure that has an orientation distribution plot or directional plot that does have enough coverage that you can trust the structure. In terms of the number of classes to use, there are 2 points there. One, if you end up asking for too few classes in ab initio reconstruction, it's generally not dangerous. You'll just end up not solving the heterogeneity, and you'll get an initial model or a structure that's the average of all the different conformations that are present. Sometimes that's not a bad thing if you have small conformational changes, and you can go to refinement, get a high resolution structure of the parts that are conserved, and then start teasing out heterogeneity with other methods. But if you're looking for wide conformational changes, that can be a bad thing. In the opposite case, when you ask for too many classes, there are two things that might happen. The best case scenario is that ab initio reconstruction ends up finding one or more classes that are actually populated with lots of particles, different conformations, and the remainder of the classes are empty. That would be great. The more likely thing, or not necessarily more likely, but a thing that can sometimes happen is that instead of creating a bunch of empty classes, ab initio reconstruction will separate one class into multiple classes, not based on conformation, but based on viewing direction. I mentioned this yesterday as well. And in that case, what you'll find is that some of the classes still look like your protein, but only from one direction. And the orientation distribution has just one directional views. And so now instead of all the top and side and front views going together to make one 3D structure, or multiple 3D structures with different conformations, all the top views end up in one class, all the side views end up in one class, and all the front views in another class. And that's unfortunately something that is a problem. In that case, changing that class similarity score can help a little bit to force all the particles to be very unsure about their classification at the beginning and try to get them to kind of all clump together into the same class. But it's also can be fixed by just asking for fewer classes. Another note about ab initio reconstruction, it is a stochastic algorithm, meaning that it's random. Every time you run it, it can give you a different result. And you can run it multiple times, and each time you run it, by default, it'll use a different random seed and produce different results. So that's a way to check for reproducibility of your structure. That reproducibility doesn't necessarily mean that the structure is correct. If, for example, you have missing views or some other problem, but it's a good idea to check. And finally, as we mentioned already, symmetry can cause issues if it is enforced by locking ab initio reconstruction into a local optimum that's incorrect. So that's everything I wanted to say about ab initio reconstruction, and we've already seen how it works on this data set. And we talked about ab initio reconstruction advanced parameters. So I think if there are any questions at this point, I can take those, otherwise move on to the next type of processing.
Perfect. Yes, we do have a few questions. So first, just going back a little bit to the theoretical foundations of what ab initio reconstruction is actually doing. Can you remind us what exactly is an ab initio class? Is it when you have multiple ab initio classes, are they multiple probable structures that are coming out from the same data? Or are we actually splitting the data into what we think are multiple conformations based on their electron density?
Great question. It's definitely the second one. We're not trying to resolve multiple possible structures from the same data. What we're trying to do, the actual mathematical model, the underlying basis for the whole method, the model says that we have K, where K is the number of classes, K different 3D densities. They're all unknown, but they're all separate. And each particle image that we observed comes from one of those density structures, not all of them, just one of them. And so when we're doing inference in this problem, what we're trying to ask is, okay, given this model where we don't know the 3D structures and we don't know the assignment of the particles, what can we say, what can we infer about what are the 3D structures that explain the data separately, and what are the assignments of particles to each class. So the output of ab initio reconstruction, if you notice, is in the form of different outputs for each class. So there's one output that says particles all classes, and that's just a convenience output in case you want to use all the particles for something downstream. But otherwise, the particles themselves are split up in the output into the different classes. So particles that fell into class 0 with the highest probability at the end of the optimization come out in this particles class 0 output. Volume class 0, likewise, is the 3D structure resolved for class number 0. Likewise, particles class 1 are the particles that went into volume, into the second class, or volume class 1, and these particles are a disjoint subset. So each particle at the end of the day is put into one bin. Realistically, like I said, all classification in CryoSPARC is probabilistic. So every particle, we're assessing it as having some probability of having come from each of the classes. But generally speaking, at the end of an optimization like this, the probabilities of the particle being in one class or the other will be quite distinct. And that's how we separate them.
Thank you. We've initialized this ab initio, or rather, this ab initio reconstruction is, oh, sorry, no. We'll come to that question a little bit later. In general, we know, for example, that in ab initio reconstruction, even with a symmetric protein, we're not actually imposing symmetry during ab initio reconstruction. In general, in the data processing, if we aren't sure about the symmetry of the given protein or complex, how can we find that out? Is there a computational way to do so?
There may be computational ways. So far, it's done entirely manually, like by eye, by looking at the refined structure without any symmetry enforced and seeing that it clearly has repeated subunits of the same structure, and then trying to do a refinement with the symmetry enforced. At the end of the day, a symmetry enforced structure has to be symmetric when it comes out of a refinement. And so if there was anything interesting about the structure that wasn't symmetric, like for example, let's say you had a overall symmetric particle where one of the subunits was a little bit different. In the C1 refinement, it's possible to see that difference. In the symmetric refinement, it's impossible to see that difference because the structure that comes out has to be symmetric at the end of the day. So you're making a trade-off. If there was some pseudosymmetry, some symmetry breaking, you're losing that information when you enforce the symmetry. On the other hand, when you enforce the symmetry, you get that many times more statistical power to resolve the structure that is conserved around the symmetry. So for example, in the case of the proteasome, or this 6-fold symmetric structure that we solved today, we enforce the symmetry, knowing that fact that we are going to enforce the symmetry. The structure we get at the end of the day is going to be symmetric, but every particle will count 6 times more than it did before in terms of signal. If there was some symmetry breaking that we wanted to observe, we shouldn't have enforced the symmetry. So at the end of the day, it's a trade-off in your biological context. How sure are you that the structure is symmetric, and how interested are you in symmetry breaking that may have occurred?
Thanks. Going back to parameters that we can enforce in ab initio reconstruction, for proteins that are encased in a micelle, would you consider changing the class similarity to a higher value? I'm assuming we're dealing with a case where we don't have further heterogeneity, and we're going, I'm not really sure what exactly the case would look like there, but let's say they're all slightly different from each other, but we have a micelle present.
In that case, I'm not sure I would do anything different with respect to the micelle. It is notable that in a lot of membrane proteins, and we'll see this a little later on with 3D variability, the micelle itself presents a huge amount of heterogeneity. So the micelle may come in different shapes, sizes, twisting, bending, and the position of the protein within the micelle or nanodisk, what have you. If it's a nanodisk, the rotation of the apolipoprotein around the target molecule. All of those sources of heterogeneity are often quite large, and depending on the conformational change you're looking for, may swamp the conformational change signal from the protein itself. That's something that can be handled in other ways, for example, with 3D variability using a mask. We'll talk about that later. But there isn't anything particularly special, I would say, that you have to do in ab initio reconstruction. If you are seeing conformational differences that are just in the micelle, and you want to force the algorithm to look for smaller changes that may be in the protein, you can of course increase that score. But it's not generically true that that will always actually do better when the micelle is present.
Thanks. And on a more practical note, can you walk us through again the differences and when you would use heterogeneous refinement, which we've said is like 3D classification in other programs, versus ab initio reconstruction?
Sure. So I guess the straightforward answer is that you often will want to use both if you're looking to resolve heterogeneity. You should first take your whole particle stack after 2D classification and use ab initio reconstruction, try it with 1, 2, 3, 5 classes and see what you get. If ab initio reconstruction is able to resolve a conformational change or multiple states that you believe are real, it's always worth taking the outputs of an ab initio reconstruction and putting them directly into a heterogeneous refinement. That will allow so ab initio reconstruction is working at a coarse resolution, and it's doing this special type of algorithm to search. But that doesn't mean it's very good at refining structures or at detecting subtle conformational changes, or it may also have some inaccuracy or just random noise in the classification of particles. So once you have initial models that are good for all the different states that you're looking for, or some subset of those, putting those into a heterogeneous refinement allows the heterogeneous refinement, which can work at higher resolutions than ab initio reconstruction, to further refine the assignment of particles. There may be a few particles that are in the wrong class after ab initio reconstruction or so on. Heterogeneous refinement will be able to improve the structures themselves by refining them to higher resolutions and make sure that the classification of every particle is correct. So it's definitely worth doing that. On the other hand, if you do ab initio reconstruction and you don't find any significant conformational changes, it's still worth doing heterogeneous refinement because there may be conformational differences within the single homogeneous, what you think is a homogeneous set of particles that are actually small, subtle conformational changes. And heterogeneous refinement can detect those, whereas ab initio reconstruction may not be able to. And so it's still worthwhile, even when you only find a single class from ab initio reconstruction, to do heterogeneous refinement starting from multiple copies of the same initial volume, to sort out and try to find smaller conformational changes if they exist.
And finally, how can we know something about the quality of our ab initio classes apart from looking at the viewing direction distribution? I think you touched on this a little bit already, but perhaps you can recap.
I guess there's two main metrics. I mean, qualitatively, you can look at the maps and kind of try to inspect their quality. And the other thing to look at is the number of particles that went into each class. Very small classes with few particles generally won't go very much further than what they did in ab initio reconstruction. Unfortunately, though, like I said, ab initio reconstruction doesn't do anything like resolution estimation. At this stage, it's not really possible to do that since we can't do two independent simultaneous ab initio reconstructions and have them actually come up with the same result. So we don't have a way to measure the resolution of the reconstruction. Instead, the resolution that we're considering is fixed as a parameter. And so there isn't really another way to absolutely verify the quality of an ab initio reconstruction other than going forward and doing a refinement and getting a resolution estimate, which it isn't too bad. I mean, in CryoSPARC, you can always just do that, and in a few minutes later, you'll know that. But that's generally what is required.
Okay, thanks.
Great. So I'm going to switch back to our slides at this point. Okay, the next topic we're going to start talking about is some more advanced techniques for doing high resolution refinement. Specifically, we're going to be talking about CTF refinement of two kinds. These methods for CTF refinement have been developed relatively recently by a number of groups. Originally, the two kinds of CTF refinement we're going to talk about specifically, one is refining the defocus of individual particles. So that means after we've done a refinement of a 3D structure, we can take that 3D structure estimate that we have and compare it against the experimental data. Up to this point, if we'd use patch CTF, for example, our CTF estimates are entirely based on the images alone, looking at those tone rings. But if we know what the structure is supposed to look like, we can always take that reference structure, project it in the direction that we know this image is looking, apply various different CTFs, different defocus values, and see which one matches the experimental image best. That's what CTF refinement is all about. It's about refining the CTF estimates based on the 3D structure that we've already computed at this point. So it's something that's done towards the end of a project, after all the pre-processing and everything else. Refining the defocus of an individual particle is something that can be very important, and especially for higher resolution and more rigid proteins, it can make a world of difference because it can directly account for the position of each particle within the ice. So with patch CTF, we're only able to estimate a defocus landscape. So different parts of the ice can have a different defocus value, but at one position, two particles can't be at different heights in the ice, according to the patch CTF model. But when we refine individual defocus parameters, we can have each individual particle having a different position in the ice. And if the particle is big enough and there's enough signal in the images, we can actually do that quite well. And that can yield significant resolution and map quality improvements. Likewise, there's a second kind of CTF refinement that we can consider doing. And this approach, these sort of higher order aberrations that we may want to correct, those are including things like that we heard about in our first day's lecture, beam tilt, coma, higher order aberrations like trefoil, tetrafoil, are all standard optical phenomenon that are known for electron beams or optical beams that describe the advancement or the phase difference in different parts of the diffracted beam coming off of the sample. They're caused by various aberrations in the microscope. For example, beam tilt is the most common one. If the beam itself is arriving at the sample at an angle relative to the optical axis, then diffraction going in one direction will be different than diffraction going in a different direction. And that causes the CTF model to be different than just a defocus and a astigmatism parameter that we previously were estimating. Each higher order aberration has its own parameter sets. We won't go too much into detail on that. This whole notion of correcting and showing that correcting higher order aberrations is actually beneficial for single particle analysis was originally pioneered by Zidanov et al. That's a great paper to read for this topic. And it talks about all the different aberration types and how their amounts can be estimated from the data. Doing the CTF refinement itself involves looking at all the particles, comparing them once again to the reference structures that we have got so far in the refinement, and computing what would be an optimal set of parameters for each type of higher order aberration to correct the presence of those aberrations that we see in the reconstructed result. Again, doing higher order aberration correction for larger, rigid, and high signal to noise ratio images for large proteins works very well. For other things, sometimes not so much. It depends very much on the data context. However, it's always worth trying. It can hurt as well. If there isn't enough signal in the data, then a lot of this refinement ends up listening to noise and actually making the results worse. So what we recommend is that at the end of every refinement, you can also try another copy of that refinement with CTF refinement enabled. And I'll talk about how that's actually done in CryoSPARC. And in some cases, that may yield a nice improvement. These methods, along with everything else we've talked about, are all important for getting very, very high resolution reconstructions. So this little video on the right that is great to look at is a 1.65 angstrom reconstruction from Empire 1000, 10,200, which is an apoferritin dataset. Apoferritin is a well-known test sample for single particle cryo-EM. It's very simple from a sort of structural perspective in that it has a high symmetry, that's octahedral, and it's very rigid, very easy to make, or not easy, but it's possible to make very good samples out of it, very thin ice, and all kinds of other characteristics that we would like. And it's easy to pick, it's circular, etc. It's just great in every way in terms of a benchmark data set for high resolution studies. Not terribly representative of what most people are trying to study in practice, but a great benchmark. And for these kind of proteins, it is possible to get very high resolution reconstructions. What's important to keep in mind in those cases is that every stage of data processing contributes to that overall resolution. A slight difference in motion correction, a slight difference in CTF estimation, particle picking, purifying your data set, 2D classification, all of those changes can yield small incremental improvements in the final resolution for these kind of data sets. Now, depending on what you're working on, the value to you between getting a 1.7 angstrom reconstruction and a 1.6 angstrom reconstruction may not be a lot, or even between 2 angstroms and 1.7, depending on exactly what you're looking for, that may or may not be extremely valuable. So it's up to each person to see how far they want to chase this high resolution possibility. It does generally take, for other samples besides the simplest ones, quite a bit of processing and playing around and data collection tweaks and so on to get super high resolution reconstructions. So coming to what we're going to try to do in practice, what we started before lunch, at least all the practical participants should have done this so far, is to import a set of apoferritin particles from the same Empire 10,200 dataset. That dataset actually has something like 400,000 particles that end up going into a very high resolution reconstruction refinement. We're actually only going to use a subset of 20,000 particles that we've prepared in advance, and everyone has imported and started processing. For that, I'm going to talk about refinement and some of the advanced parameters. We actually covered most of the advanced parameters in refinement already, but I'll talk about a few more that we haven't touched yet. And then we'll see how to do CTF refinement as a standalone process in CryoSPARC, and then CTF refinement on the fly as part of 3D structure refinement to make it very straightforward and easy to apply. We'll also talk a little bit about exposure groups. So in your CryoSPARC project, earlier before lunch, you should have created a workspace, called Apoferritin or whatever you want to call it. And there you should have imported the data, imported a volume for an initial model, and then run 2 refinement jobs. I'm just going to show the results in my own instance here. Okay, so the 2 refinement jobs that we started, and this is now using the instructions that are listed here for Apoferritin. The 2 refinement jobs that we ran are different in a simple way in that one of them doesn't use CTF refinement and the other one does. The first one, first of all, I should mention that these particles, originally we processed them in CryoSPARC and we used patched CTF estimation, which meant that each particle had actually its own defocus value based on the defocus landscape from patched CTF. And it turns out that that's already a very good CTF estimate. So with those CTF estimates, we were getting extremely high resolution results already, and the difference after doing CTF refinement was minimal, especially for the smaller subset of particles. So instead, what I did when preparing these particles is that I fixed them all to have a constant defocus across every micrograph. So that's similar to the assumptions of other older techniques for CTF estimation where the image is assumed to be flat. So starting with that as our baseline, doing a refinement of these 20,000 apoferritin particles, which obviously are of extremely high quality and a very good sample, gets us to a refinement resolution of 2.3 angstroms. That's obviously already very good. But we can go further, and the way we can go further is by doing CTF refinement. So the second job that everyone has run implements and uses CTF refinement during 3D reconstruction, and that reaches a resolution of 1.9 angstroms after CTF refinement. So that's a pretty big jump from correcting for these defocus and other higher order aberrations. To actually get into what's happening there, I'm going to talk about the 2 standalone job types in CryoSPARC. You can create these by going to the job builder, although you don't need to actually run them. Go to the job builder and look for CTF refinement jobs, and you'll see that there are 2. There's a local CTF refinement job and a global CTF refinement job. And they implement the 2 kinds of CTF refinement that I mentioned, one of them being the defocus of every particle, so that's local, and the second one being higher order aberrations that can only be seen in aggregate across the dataset. So the local CTF refinement job, what it does is it takes in particles and a 3D structure, in fact, 2 half maps from a 3D structure, and then it uses those 2 half maps to compare for every particle image the projection from this 3D structure against that individual particle image. And it does that comparison again at a bunch of different simulated CTF values. What we get is that for each particle, we compute one of these landscape type diagrams. So let's look at this one in the middle. That's for a particular single particle. And the values on the x-axis are, sorry for the mashup of the numbers, but the values on the x-axis are the relative defocus compared to the current estimate of the defocus. So if we previously thought that this particle is at, let's say, 2 micron defocus, then each of these points on the x-axis is plus or minus in angstroms from that initial defocus estimate. The line that we're measuring here is the log probability or level of discrepancy between the particles between the experimental image and the simulated projection at these different defocus values. What we should see is that at some point, this curve takes on a minimum, and it may not be at a defocus change of 0. That difference, that position where the discrepancy, the matchup between the experimental image and the projection is best, where it's minimized, that's actually the optimized per particle defocus that we wanted to find all along. So if you look at these curves, you see that sometimes it's a little bit more than 0, sometimes it's a little bit less, and each particle gets its own individual refined defocus value. Together, those are aggregated. There's lots of them plotted here. Those are aggregated into this histogram, and we can see that lots of particles have an updated defocus as much as plus or minus 500 angstroms away from where we thought the particle was. And that's actually literally tracking the height of the particle in the ice. If the ice was only 1,000 angstroms thick, then we're looking at particles moving from like particles at one surface or the other surface of the ice, or anywhere in between. This is also capturing the fact that the ice may not be flat. So our model so far, our previous defocus values that we plugged in here were all flat. All the particles had the same defocus. But if the ice was tilted, then we would see the individual defocus of every particle shift to match that tilted plane and so on. So that's what is happening in this local CTF refinement job. The global CTF refinement job, on the other hand, is also taking in the 3D volume, 2 half maps, and all the particles, again, comparing every particle image to the 3D structure. But instead of looking for individual particle defocus changes, it is looking for systematic aberrations that are present when comparing the experimental images to the projection of the structure. And those aberrations are, I won't get into the details of how you can tell that they come from a certain kind of electron beam aberration or electron beam phase shift issue, but each of them corresponds to a certain kind of phenomenon in the electron beam. So there are 4 different kinds of aberrations that we can estimate and correct for in CryoSPARC: beam tilt and trefoil, which are both odd first and third order aberrations. We can also correct for spherical aberration. We can re-estimate the spherical aberration and correct for tetrafoil, which are both 4th order aberrations. So this data set is not a great example. It doesn't end up having actually too much aberration left to correct. But in other data sets, you can see very strong aberrations. If I just go back a couple slides here, very strong aberrations that end up creating these patterns that what are called Zernike polynomials that account for the phase shift in different parts of the diffracted beam. So once those are corrected, resolutions can reach much higher. What's interesting is that in CryoSPARC, instead of having these just be standalone jobs that you can run and then have to take the particles from these and rerun a refinement using those one-time updated defocus and CTF parameters, instead, within the refinement job, this refinement new job, which all the practical participants have run, we have options to turn on per particle defocus optimization and per group CTF refinement within the refinement cycle. And that means that in this job, if we look back a few iterations, at iteration 5, for example, we are actually doing the same CTF refinement at this point in this refinement cycle. So at iteration 5, we start refining the CTF. The defocus values are updated, the astigmatism and other higher order aberrations are estimated and corrected. And then we do another round of refinement to update the angles and poses of all the particles. And then we re-estimate all the defocus values and update them again. What you see here is that in the first time that it was done, particles moved quite a bit in their defocus values, up to 500 angstroms. The second time it was done, particles moved by a smaller amount, but still some amount of change in their defocus. And these small changes are due to the change in the 3D structure that updates at every iteration and the change of the angles of the particles that are also being refined. So jointly refining all of them can sometimes give a slightly improved result. And at the end of the day, the result of this refinement job is a fully optimized CTF refined high resolution 3D structure. And as I showed before, in this case, with just these 20,000 particles, we can get to 1.9 angstrom resolution. So that is everything about CTF refinement and ultra high resolution data. The last thing I want to talk about is exposure groups. A lot of times with very high rate image collection, a strategy that's used is that images are collected by shifting the beam to multiple positions, different holes at the same stage position for the sample. And what that means is that in every position of the beam shift, the beam aberrations can be different. And so the data set needs to be cut up into those sections, each section being one set of images collected at the same beam position. And then CTF refinement for those needs to be done all separately. And I won't go into the details of how to do that, but we have a long and very detailed guide at cryospark.com/docs in the tutorial section for CTF refinement. It covers everything I talked about for local and global CTF refinement, as well as a section on how to cut up your data set into exposure groups if it was collected in the manner that I described, and then refine the CTF parameters of each of those separately. It's pretty straightforward, and there's a utility job that makes it even easier to do in practice. Okay, I think this is another natural point for any questions, if there are any from the preceding sections.
Yes, perfect. So overall, I think the flavor of questions regarding CTF refinement start off with, when exactly in my data processing workflow should I even be considering doing CTF refinement? What do I already have to have in order for this to be useful?
Great question. I think the answer is actually pretty simple. You can start considering doing CTF refinement anytime you're doing a refinement of a structure, when you're actually at the point of doing a homogeneous 3D refinement. So that means you've already separated your particle stack into different conformations, classified everything as much as you can, or maybe not as much as you can, but you're at a point where you're doing a refinement. You can always do that refinement without CTF refinement, and you can do that structure refinement with CTF refinement. Like I said, it's not safe to assume that CTF refinement will always create an increase in resolution or improve the map. It is totally possible for it to make things worse if any of the assumptions behind CTF refinement are broken. Earlier when I showed the case study example of the NAV channel protein, that's a great example. Unfortunately, I can't show you the refinement jobs. I don't have them ready to show, but it's a great example where if you use the refinement, if you use the defocus estimates from patch CTF, you get the results I showed. If you try to do defocus refinement or any kind of CTF refinement, it actually makes the results worse because the protein is flexible and the micelle is disordered, and it's a small protein in difficult conditions, which means that the reference structure when we're doing CTF refinement never matches the particles very well. Either the experimental particle is in a different bending position, or the micelle is in a different configuration, or something else is off that makes those two not match up, and it throws off this defocus refinement and CTF refinement completely to the point where the results get worse. So in general, it's always better to try refinement without and try refinement with CTF refinement.
Great. We in our tutorial here have done CTF refinement within refinement. If I'm using standalone CTF refinement jobs, on the other hand, do they need to be done in a certain order, i.e. first do local and then global?
That's a good question. In theory, that could matter, yes, because the optimizing the local and global CTF parameters has some interplay, where if you change one of them, it could change the optimal value for the other one. We haven't seen that matter too much, but I would say it's best to do local CTF refinement first, because the defocus is a much more noticeable change than the aberration correction in a lot of cases. So it's better to get the defocus as right as possible and then try to optimize the aberrations. In general, though, unless you're doing something really strange, it's better to just use the versions of this refinement that are built into the refinement job.
Okay, some more questions regarding when CTF refinement might be useful or might make a difference based on your actual data set. So is there any relationship with the size of the particle where you would expect to see more or less of an improvement by using CTF refinement?
Absolutely. The relationship is actually to do with the signal to noise ratio overall. And the size of the particle often is a major contributor to the signal to noise ratio. If you consider a case where you have ice of a certain thickness and a certain amount of electron dose, the smaller your particle, the less of the electron diffraction is happening in the protein versus the ice. And the less signal you're creating by that electron diffraction due to the protein versus noise in the detector. So with everything else being equal, the particle size exactly modulates the signal to noise ratio. When you have a large particle, there's lots of signal. You can easily detect the difference between a particle that has a defocus of 10,000 angstroms versus 10,001 angstroms. Maybe not that precisely, but you can easily tell the difference between a particle that's moving a little bit up and down within the ice if it's large and rigid. If it's not, if it's small and your ice is thick, then there's not really much hope. However, with small particles in thin ice, it is still possible to get an improvement from CTF refinement because the thinner the ice, the better the signal to noise ratio will be.
Great.
One question, at any of these stages, are you running into the problem of the defocus difference or treating the defocus difference between the top and the bottom of a very large molecule, as David discussed yesterday?
That's a great question. No, so far none of the methods we've presented deviate from the infinitely thin sample assumption, in the sense that all of our reconstruction techniques in the math involved assume that an individual particle is infinitely thin relative to like the small angle scattering assumptions for the electron beam. So we are not considering the fact that the top of the particle and the bottom of the particle are at different defocus levels. That is something that could become important for very large particles, or in other cases like lower energy microscopes, but at 300 keV for the kind of standard size of particles that most people are dealing with, it has not yet been shown, except at extremely high resolutions, to be an important factor.
Relatedly, is there a certain resolution threshold at which it makes sense to do CTF refinement? Like, let's say you've already refined a structure to 3.5 angstroms, or perhaps it's in more of the 4 to 5 angstrom range. Mm-hmm. Is there any sort of consideration there for whether or not it makes sense to run CTF refinement?
No, if the quality of the data is good, then even from 4 or 5 angstroms, CTF refinement can start to make a difference. And if the higher resolution you already are at, the more confidence you have in the quality of the data, so the more, from a prior belief sense, you would expect CTF refinement to help. But again, I would say that in practice, all these questions are not what you should be asking. You should just try it, and that will answer the question definitively.
Great. A couple more questions regarding exposure groups. If my data was collected with beam image shift, would running CTF refinement during homogeneous refinement, as we've done here, be a bad idea before splitting into exposure groups? Should I first split into exposure groups, and then run standalone CTF refinements and then recombine them again for a homogeneous refinement? And a related question is, dealing with homogeneous versus heterogeneous data. Should you be actually doing CTF refinement on every single different class of structures, or should you be doing it at the same time?
Great. I'll try to answer them both. So with exposure groups, if you haven't yet split your data into exposure groups, then you shouldn't try to do global CTF refinement, which is aberration correction, since the aberrations in the beam are going to be different in each case, and putting all the data together is going to make a mess of things. You can do local CTF refinement perfectly fine, regardless of the exposure group, since the defocus doesn't have this notion of being different in terms of how it needs to be treated between the exposure groups. All types of CTF refinement, the aberration correction, global CTF refinement, whether it's standalone or inside of the refinement job, respect the exposure groups. And so if you do split up your data into exposure groups, you don't have to run CTF refinement separately. You can just throw all of that into a refinement job, turn on the higher order aberration correction, and let it be. So I think that answers the first question. The second question, in terms of heterogeneity, yes, you should always do CTF refinement separately after classifying as much heterogeneity as you possibly can, because as I mentioned a few times, the basic assumption of CTF refinement is that the experimental image actually contains exactly the same thing as what's in your reference structure to a very high resolution. So if you have heterogeneity of any kind, that's going to break the assumptions of CTF refinement and make it not be able to detect the right CTF value. So you should split up your data into as many different conformations as you can possibly find, and then refine each of those separately with CTF refinement being done individually. And this is conjecture at this point, not definitively proven, but I believe that that's the case even for small conformational changes. Like for example, the open and closed version of an ion channel are different enough that CTF refinement will fail if you put them together.
Great, thank you. I think that's all of our questions on CTF refinement.
Excellent. Just how are we doing in terms of time before the next scheduled break point or anything?
The next scheduled break is in approximately 20 minutes.
Okay, perfect. So what we're going to do next is move on to some interesting new kinds of refinement methods to deal with particular kinds of data. The next one is a particular favorite of mine, but we're going to be processing a subset of data from Empire 10,288. This is a GPCR protein dataset. Originally in our plan, we were going to actually start with the raw movies and do the whole data processing, but due to how much time we had today and the changes in plans, we're now going to start with particles that have already been picked and classified and extracted and everything. Not classified, but just picked and extracted and curated to some degree. And those particles we are going to import into CryoSPARC and then do some 2D classification, do an ab-initio reconstruction, and then do two kinds of refinement. Our standard refinement that we've been using so far, and a new type of refinement that I'm going to be explaining in some detail called non-uniform refinement. Now, unfortunately, it's plausible that these refinements won't actually finish by the time we finish talking about how it all works. So we might have to come back a little later to look at the results, or I can show you what they look like with a version of this that's already finished. But let's just dive right into CryoSPARC, import the data, and get our processing going. So in your own project, please create a new workspace. You can call it GPCR. And in that workspace, create a new job, which is an import result group job again. So once again, we're importing a CryoSPARC result. So it's just a single file that we have to point to. And as I mentioned before, this job can be used to import any kind of CryoSPARC result into another CryoSPARC instance. The only caveat is that the data that you're importing has to be inside of the project directory where you are importing it. And that's on purpose because we want to make sure that project directories remain fully self-consistent and self-contained. So in our case, we've taken care of that by creating some helpful symlinks. So if you go to the /scratch/training/projects directory, you'll see a bunch of project directories, and one of them will have the same number as your project number. Mine is project 6, and so I'm going to choose project 6 here. And inside of my project, I will see a Slack workshop data folder that's actually a symlink to /scratch/training/Slack workshop data. Entering that directory, and so I'm still inside of my project folder, but inside of the workshop data folder, I'm going to now go ahead and open up the GPCR folder, that's dataset number 2. Open the particles directory and find the CSG file. And that single file contains all the metadata information about where all the other files are and lets me load in this result right back into CryoSPARC seamlessly. So selecting that, I can now queue that job and let it import my particles. Just another note, as we import these particles, you'll see that the job reports where it's importing from, and it also reports what kind of results it was able to find. So it's found one particle CTF result for all these particles, and particle blob. Blobs refer to the actual raw data on disk. So it's found raw data and it's found CTFs. That's all we need at this point. The next thing we're going to do is create a 2D classification job, taking all of our particles, 123,000, and plopping them into our 2D classification. We'll just run that with default parameters, and you can go ahead and launch that. I'll switch over to my own instance where I've already done so. This 2D classification, in my case, took under 10 minutes. So we'll just let this run behind the scenes, and we'll come back to select some good classes. But what we can do to save some time later is create the chain of jobs that we want to run right now. So going back to your project where you have your 2D classification job now launched, you can open up that job and you'll see that it has particles and class average outputs. What we'll do is go to the job builder and create a new select 2D job. Connect the particles and the class averages from our 2D classification to that and queue that job. That job now is going to be waiting because its parent is not finished yet. So it's going to be waiting. But then we can take the output of that select 2D job and plug it into an ab initio reconstruction job. And again, here we're going to use default parameters, a single class, and just queue that job as well. This is an example of what I mentioned yesterday, that it's possible to construct an entire workflow of processing steps without having actually finished all the previous processing. Now this ab initio reconstruction job, we're going to take its output and feed it to a homogeneous refinement, new. So we want the particles. Since there's only one class, well, we can use particles all classes and the volume, and with all default parameters. And finally, now we will create a non-uniform refinement. That's a new job type. It looks almost exactly the same as a regular refinement. It has a bunch of extra parameters, and it does something different under the hood, which I'll talk about. For this one, we're going to take the outputs of ab initio reconstruction again, not our first refinement, but the ab initio reconstruction, all the particles and the first and the volume. And again, default parameters, and we can queue that as well. So this is what you should see in your project now if you're a practical participant, except that your 2D class job should actually be running. I didn't actually start mine to not use up resources that were allocated to others. And the other jobs in your chain should be ready to go as soon as this 2D classification is finished. So we'll come back to this to select some 2D classes and let the rest of the processing go on. But I'll just show you what things look like in this other workspace where I've already completed that same processing. So our 2D classification job yields some 2D classes. And if we zoom in here, we can see that there are some great side views where we can see directly the secondary structure down to transmembrane helices in our GPCR complex from a lot of side views that are maybe slightly different viewing directions, but not very different from each other. And a few other types of views, some front views, very few in the way of top views, but everything kind of looks like a particle. There are some of these weird things that look like maybe two particles that are on top of each other. We probably want to exclude those. Here's another example of that. And then there's some things that are mispicks, like that's not really our particle. Not sure if that's our particle. So the classes are a little more dirty than the data sets we've worked with so far. And cleaning this up actually is really important. If you were processing this data from scratch, this would be a prime case where you might do multiple rounds of 2D classification, even do an ab initio reconstruction with multiple classes just to see whether you get some junk classes you can throw away, and use all the strategies that we talked about to clean up this particle stack. If you're aiming to get a single high resolution reconstruction. So far, we don't really have any indication or suspect that there are conformational states that we really want to investigate at this point. So we're not worried about that right now. We'll find out later that was actually not a good assumption, but so far we don't really detect anything that hints that there are multiple conformational states that we're looking at. So we can let all that run, and our ab initio reconstruction should recover an overall structure of the GPCR complex. You can see that the viewing distribution is not great. There is a wide band of different views, but there's a huge number of particles that are all kind of this sort of front view according to this orientation. That's not a bad thing, since they do have lots of diversity in terms of their angles, but it's not a perfect orientation distribution. It's good, not best. Okay, so coming back to our slides, what I'm going to do, as long as the practical participants are able to follow these instructions and start off those jobs, and that can be happening in the background, I will start talking about non-uniform refinement, what its sort of purpose is, conceptually how it works, how it really works under the hood, and what kind of results you can get out of it. It's an algorithm that has become very popular. It has been used in many high-profile membrane protein structures, even in some of the latest coronavirus structures of the spike protein. In general, it is a technique that, by the way, I would recommend anyone who's interested read the paper that we have right now in bioRxiv, non-uniform refinement. In general, the purpose of the method and the results that we were able to get show us that accounting for spatially varying rigidity, order, disorder, and other structural properties across a protein can significantly improve particle alignments and therefore the resolution and map quality that we can get. It turns out that although this is a general principle, experimentally we see that the cases where this is most true is where there is lots of spatially varying rigidity, order, and disorder. And the best example of that are membrane proteins. When there is a micelle nanodisk or floppy fabs attached or any other weird scenario, non-uniform refinement can often make a big difference. I'm going to dive in a little bit into the motivation behind the method and exactly why we came up with it or why it works. And it's actually been available in CryoSPARC for, versions of it have been available in CryoSPARC for about 2 years now, and we just finally got around to writing the paper. So what non-uniform refinement is based on is the hypothesis and observation that in a standard refinement, the way that we've looked at refinement so far, one of the key principles that I talked about a little bit earlier was regularization, which is the process of making sure that we don't overfit and that all of our estimation of different things, whether it's the 3D structure or the alignments of the particles or whatever it is that we're estimating, even CTF refinement, is driven by real signal and not by noise. As soon as we get into a territory where things like particle alignments are driven by noise, they can all, over iterations of refinement, start to accumulate error that reinforces itself, and overtakes the real structure of the protein in driving those alignments, destroying our ability to resolve the structure of the protein. We don't see, thanks to the methods that I talked about earlier, like the gold standard FSC technique and using FSC for regularization, we don't see overfitting in such a drastic way anymore these days. But it turns out, and what we were able to discover and show with this project is that in many cases, there is still detrimental overfitting that is happening, or underfitting, which is the opposite of overfitting, that ruins the alignments of particles and ruins our ability to resolve high resolution structure that we otherwise could have gotten from the same image data. So coming to that point, in a standard refinement, our regularization technique, and in fact, all the operations that we do are grounded in the Fourier basis. It's very natural if you're a crystallographer or any other kind of structural biologist to think of a protein structure or an atomic structure of any kind in its real space form, where you have electron density or whatever it is, a Coulomb potential, whatnot, atomic coordinates in real space. And then you have the dual of that related by the Fourier transform, which are the structure factors, or literally each Fourier component representing a spatial plane wave of density, a spatial frequency of increasing and decreasing electron density. Together, adding up all of these plane waves makes up this density structure. Literally, when we're doing a Fourier transform, we are changing the basis in which we represent the 3D structure. Instead of representing it as a bunch of voxels independent, each one with an electron density, we're representing it as a bunch of electron density waves that when added together make up this 3D structure. And in Fourier space, lots of things become trivial. First of all, we get the Fourier slice theorem that connects in Fourier space the Fourier transform of 3D structure to the Fourier transform of a 2D projection image that we actually collect in the microscope. So that's really good. CTF correction is also trivially straightforward in Fourier space because the CTF is a multiplicative operator in Fourier space, whereas in real space, the CTF would be much more complicated and difficult to work with. We also get the ability to do 2D to 3D back projection. So we can reconstruct 3D structures in Fourier space very easily. And we can measure the Fourier shell correlation, which we talked about earlier, by just isolating shells of Fourier components that correspond to plane waves of the same frequency in different directions and comparing those between maps. And that's the basis for how we do regularization in regular standard refinement. But the problem, or one property of this idea, is that each of these Fourier components, which we regularize using FSC or whatnot, each of these Fourier components represents a plane wave of electron density that goes across the entire molecule and is totally homogeneous in terms of what it looks like from one end of the molecule to the other. So when you do operations in Fourier space, like regularization, they are almost always going to be spatially uniform operations. Simple operations in Fourier space are spatially uniform operations in real space. And that's actually the whole problem that we're trying to get around. The key intuition in non-uniform refinement is that regularization in the Fourier space basis just doesn't make too much sense when you have a real life molecule that has lots of problems. So this is an example of a TRPA1 ion channel, Empire 1024, a regular standard refinement result. And it's colored by local resolution value just to give a sense of the different regions. And the threshold when we were looking at the map is set relatively low so that we can see all the different parts of it, even the low density parts. What we see there is that there are some very distinct different regions. There's a sort of core region that's in red, that's sort of rigid and well ordered. There is a micelle, detergent micelle, light blue, very disordered and only resolvable at a lowish resolution. And that's where the membrane, where the protein would be in the membrane. There's also a totally flexible floppy tail that is completely washed out and just turns into this dust. All of these regions are examples of places, are examples of different parts of the structure that have different properties. But when we do regularization in Fourier space, we're explicitly assuming that the structure is the same in terms of its properties that we are regularizing across its spatial extent. And that's exactly what is the problem. So in non-uniform refinement, what we did is to simply change the regularization operator in a refinement. Everything else is the same gold standard split, same resolution estimation, same way of doing alignment and so on, branch and bound, everything. But regularization, instead of happening in Fourier space, should happen in a different basis that is adaptive over space and frequency. So different parts of the structure can be represented and regularized in different ways when we are talking about a refinement. How that's actually done under the hood, if anybody's really interested, I would recommend reading the paper. We spent a lot of time writing out the math and making it very clear how the technique works. It's actually pretty neat in that we were able to formalize this whole idea into a notion of cross-validation optimization. So instead of just kind of taking an ad hoc approach and saying, well, we know we want to kind of look at different regions of the particle and try to isolate which ones are different from each other. Instead of that, we decided to come up with a sort of formal framework from which we could derive an optimal regularization that would give us the properties that we were looking for without any tuning or creation of masks or parameters to choose. And it turned out we were able to do that in a pretty nice formulation where we set up the problem as one where we have to optimize parameters of our regularization operator. And to do that, we compare the two half maps within a refinement. And I'm skipping over some detail here, but we compare the two half maps in a refinement. We filter one of them and compare it to the other, and then we filter the other one and compare it to the first independently. And by doing so, we can tell what kind of filtering, spatially adaptive filtering, would correctly keep the information in each part of the protein that's actually relevant for alignment of particles and remove all the noise and overfitting. And what that looks like in practice is something like this. So if you do a reconstruction without any regularization at all, you end up with a result that is very noisy. So all the solvent has tons of noise. This is a slice through a membrane protein, by the way. So you can see this cloud of density that is a slice through the actual micelle in this case. And then there's protein density where you can see sort of side chain details and slices through alpha helices and whatnot. Without any regularization, the structure, there's noise everywhere, and in the solvent, in the micelle, in the protein, and different parts are all treated the same. Similarly, in uniform regularization, which is based on the common techniques that we mentioned before, FSC, all parts of the structure are treated the same. So we've removed some noise from everywhere that was commonly present across the board, but there's still lots of noise left in the background, lots of noise left in the membrane micelle. Only the protein structure looks a little bit cleaner than before. With spatially adaptive cross-validation optimal regularization in non-uniform refinement, what ends up happening is that, again, without any masks, without any tuning or hand setting parameters, we're able to realize, the algorithm is able to tell that the solvent shouldn't have any information in it at all. The micelle is so disordered that it shouldn't have any details either, just this general sort of shape. And the protein density should remain perfectly intact with all of its high resolution detail present. And that is actually the crux of non-uniform refinement. Doing this kind of regularization at every iteration of refinement in a way that doesn't break the gold standard split, and there are a bunch of other details to make sure of, and doesn't cause overfitting of its own, that can all be read in the paper, yields some really dramatic improvements in the resolution we're able to get for membrane proteins. So this is an example from our paper. This result was actually published in the original structural study where they used non-uniform refinement before we were able to write our paper up, but it's an astonishing case of an improvement due to this algorithm. The protein itself is a plasmodium falciparum transporter, PFCRT. I don't know too much of the biology. I know it's somehow involved in chloroquine resistance for malaria. And it's a small membrane protein, quite small. Like the entire membrane protein is actually fully inside of the membrane, and there's a fab bound, that's one fab attached to the protein, so very small. And it was very difficult to purify and handle apparently, which is why there's only a small number of particles that were available, something like 17 or 17,000 particles, I believe. Without non-uniform refinement, using just standard refinement techniques, in whichever way they're implemented, yields only a 7 angstrom reconstruction in this case. Oops, sorry. Yields only a 7 angstrom reconstruction in this case. Using the exact same data and everything else exactly the same initial model, same everything else, no tuning or masks, non-uniform refinement yields a result at 3.6 angstroms. And that's a huge jump where basically the structure goes from being not really intelligible to perfect sidechain detail, great for building a model. And obviously this is an extremely dramatic example. You shouldn't expect this kind of thing to happen in every case. I mean, it's worth trying, but what was true in this protein, in this case, is that the micelle density relative to the protein was very large. The total amount of mass in the disordered regions of the structure, which is the micelle, were very large compared to the protein itself. And that meant that in a uniform refinement, the effects we were talking about were very strong, and particle alignments were all becoming systematically wrong because of misregularization. What happens between uniform and non-uniform refinement, the actual real reason for the change is that the particle alignments become much better once we start regularizing the structure properly. And we can see that here. These histograms show us the difference in pose angle between the regular refinement and the non-uniform refinement. And you can see that on average, many of the particles change their pose by something like between 6 and 20 degrees, which is a huge amount of pose change, obviously. And for such a small particle, that makes a lot of sense. When the structure reference is incorrect and things aren't going well, it's very easy to get the orientation of the particle completely wrong. But with non-uniform refinement, that problem goes away, and both shifts and rotations are made much more accurate, resolving all this extra detail. So I'll just show some videos for fun of the difference before and after. Sorry, that's before, and that was after. And these are also the same. Sorry, these shouldn't flip so fast. Before and after electron density maps, meshes around alpha helices and beta sheets, where obviously in this case, the difference is night and day. This is another example, also from the paper, and this is another example we talked about previously, the NAV 1.7 channel. In this case, the protein is larger and the relative amount of mass in the micelle is less, but there are two fabs that we've seen earlier are very floppily attached. And the overall structure also has a lot of flexibility. And in this case, this is another case where non-uniform refinement makes a difference between overall resolution of 3.4 down to 3.1 angstroms. And the improvement in the map quality is also quite significant. I'll just show these videos first. Before and after on the right-hand side, and some detail views. Before and after on the right-hand side. And these are the a few helices before in black, after in purple, where you can see that the side chain resolution, ability to resolve rotameric conformations of side chains, and even resolve some side chains like on the left, right, that couldn't be resolved before, is greatly improved. So that's non-uniform refinement. And the next thing we want to do is, probably by now, our GPCR protein hasn't finished processing for most practical participants. But in the interest of time, what I'm going to do is just explain the jobs and what we've done here, and the difference in the map quality and the difference in the resolution here. So this GPCR was a good example where we saw that, we can see that in this example, we did a single ab-initio reconstruction, started with as an initial model for both types of refinement, and the standard refinement made it to 3.8-ish angstroms. This was an example where the masking difference between the very loose mask and a tighter mask was also dramatic. But this uniform refinement only made it to 3.8 angstroms. And you can see in the slices of the 3D density that there's lots of structure in the micelle that really doesn't make a lot of sense. And that actually is all overfitting or underfitting or spurious density that is not actually intelligible, not really trustable from the signal in the data. And that's what's causing the misalignment of many of the particles. Non-uniform refinement, on the other hand, has a density map where the micelle is very much more smooth, and the overall resolution reaches 3.4 angstroms, which is obviously a big jump. And the FSC curve is overall higher as well. We can see also the orientation distributions, again, just verifying what we saw from the Ab-initio reconstruction. Not very good, and you can see from the posterior, this second plot, that there are regions, some regions have a precision up to 1800. These units are actually not something that you can transfer from one data set to another. They're really an arbitrary unit axis, but you can relatively tell the difference between different orientations or different positions in Fourier space. So some parts have a highly sampled Fourier space and some parts are much less. But it turns out that's not such a big deal for this protein, and we still get a 3.4 angstrom result only from 300. So these particles, I should have said, are a subset of the full data set. The full data set has some 400,000 particles that make it into the refinement and gets to around 3 angstroms or 2.9 angstroms. So far, we've only used a subset of 300 movies out of the full 2,500 movies in the data set. And so these first 100,000 particles are just from that small subset. So what I'm going to do is actually show the difference between these maps. I'll try to open them both up. That'll just take a few seconds to download. And this one, I'm going to open the sharpened map in both cases. And we'll see what it looks like. Sorry, just give me a moment. Great. So the first one that is opening up is our, the first one that's opening up is job number 44, which is the non-uniform refinement. I'll try to also open up the other one. Number 34. Sorry, just bear with me a moment. Project 34, job 43. Okay. So in these two reconstructions, the yellow is, which I'll show first, is the uniform refinement of the structure. Again, this is with a small subset of the data, so we don't expect the refinement to be perfect anyway, but we can see that there's lots of density and structure in the micelle. It's not really ordered structure, it's all just noise. And that's exactly what the problem ends up being, that not only in the micelle, but in various other areas of the protein, you can see also all kinds of excess density that really shouldn't be there at lower thresholds. Parts of the structure are being misregularized. So that's a standard refinement, and that's what our transmembrane helices look like here. Right there, yeah, 43 and 44. So on the other hand, if we look at our non-uniform refinement, we'll see that now the overall reconstruction quality, still not great because it's a small subset of the particles, but when we compare them, let's say we look at some regions of the structure that are, we'll find out actually later that the transmembrane domain here is highly flexible, and that's why it's washed out, especially with a small number of particles. But if we compare these two at a similar density, we see that the non-uniform refinement has been able to resolve substantially more detail in many parts of the structure than the standard refinement. So practical participants can feel free to go ahead and also make that comparison and download the two maps and see them. Often with non-uniform refinement, we'll find a substantial improvement in the overall resolution of the structure, but it's important to remember also that FSC measurements are naturally a global resolution measure, and that's how we do validation. So when we see an overall improvement in the resolution of the average structure, it could mean that some parts of the structure have improved a lot and other parts have stayed the same or gotten worse. And that's actually what happens in non-uniform refinement. Some parts of the structure in terms of their map quality and resolution can improve very dramatically, and other parts may stay the same or get worse. And the overall resolution improvement is what we see in the FSC. Okay, I think that's everything I wanted to say about the GPCR results and non-uniform refinement. So I think it's a good time for any further questions.
Sure, we can ask our non-uniform refinement questions, and I think we have a scheduled break. So first off, can we use non-uniform refinement with the outputs from homogeneous refinement?
Yes, non-uniform refinement actually, as I mentioned, does everything that a regular refinement does. So you don't need to start it with an already refined model, which is why I showed here that you can just go from ab initio straight to non-uniform refinement. Non-uniform refinement does the full global pose search and everything that a standard refinement does. The main difference is how the regularization is done. And so it is possible to start non-uniform refinement with any input of an initial model and particles. They don't already have to be aligned, but you can take the output of a standard refinement and continue a non-uniform refinement.
Similarly, can we apply non-uniform refinement after a heterogeneous refinement? So where we have multiple classes.
Absolutely. You can treat non-uniform refinement exactly the same as a standard refinement in the way that you would use it. In fact, for many people, if you're not too concerned about run times, you should pretty much always use a non-uniform refinement because we have never seen any cases where it actually makes things worse, but it does take longer, sometimes 2 or 3 times as long as a standard refinement. So in many cases, you may be better off just always going straight to non-uniform refinement from even any kind of ab initio or heterogeneous classification scheme or method that you have, straight to non-uniform refinement, especially if you're working on a membrane protein. Probably all of your final refinements that you compare from different classes and so on will be non-uniform refinements, so you may as well start with that.
Relatedly, is non-uniform refinement more computationally expensive than a homogeneous refinement as a rule?
Yeah, definitely. In the current implementation, it is. We actually have an updated version of non-uniform refinement that's going to be coming out soon. This paper that we wrote is under review, and at the time when it's published, we will be releasing that new version as well. That should be a lot faster, and in some cases also works better. It also has fewer parameters that you can sort of change that break things. It's a little more robust in that sense.
In terms of the size of molecules that we can work with, let's say we're working with a small membrane protein, approximately 60 kilodaltons, where it's completely inside the micelle. Is that impossible to process?
It all comes down to the ice thickness. 60 kilodaltons is really at the small end of what anyone's been able to show, even for globular proteins, non-membrane proteins with cryo-EM ever, at least with standard hardware. Actually, yeah, pretty much ever. So it's going to be difficult no matter what. But if the ice is thin enough, a membrane protein that's 60 kilodaltons is actually substantially easier to work with than a globular protein of 60 kilodaltons. The reason being that the micelle itself at low resolutions contributes a huge amount of signal to being able to align the particle. We've had at least one case, and it's not published yet, but some results, partial results that we were allowed to show are in our non-uniform refinement paper of someone dealing with the exact same case. It was a trimeric protein that has threefold symmetry that's fully encased inside the micelle, and it's 90 kilodaltons. So not quite as small as 60, but in the ballpark somewhat. In that case, non-uniform refinement again made a huge difference, but the thickness of the ice was really the crux of being able to do anything at all. It was only once they were able to prepare a sample with very thin ice, and in that case, the micelle helped a lot in aligning the particle overall. I mean, in that case, you have a lot of problems where if you just have a micelle and the protein is embedded inside, if the protein doesn't have a lot of structural features that are really unique, it's very easy to get the orientation of the whole thing wrong by 180 degrees. And you also have the rotation of the micelle. And if the micelle itself has any variability, that might create more difficulty in alignment than the protein has signal that can drive the alignment. So it's a very difficult scenario, but I wouldn't say it's categorically impossible. Just you have to be very, very lucky.
Let's say that you're working with a protein that has a flexible region, which you think can actually break the symmetry overall. Should you then impose C1 symmetry in non-uniform refinement rather than a different form of or different type of symmetry?
This is again a sort of depends on the data set and the problem, the protein itself. If the question is that you expect that one of the subunits or some part of the protein is going to break the symmetry, the only way you're going to find it is without enforcing symmetry. With one caveat that we'll be discussing in the next section, 3D variability. It is possible so non-uniform refinement doesn't have a lot of interplay with this issue. It has the same exact characteristics in terms of symmetry as a standard refinement, which is that if you enforce the symmetry, there will definitely be symmetry in the output. And if you don't, you will have much less signal to deal with. But I'll just say shortly what I'll be saying later in more detail. What you can do is refine the structure with symmetry to get the highest resolution you can, and then use 3D variability to separate the symmetric and pseudosymmetric conformational changes, which can be detected with that method that I'll show a little bit later.
Thanks. Previous to this section, we talked about CTF refinement. Can you do CTF refinement and defocus refinement in non-uniform refinement?
Great question. No, right now the current implementation of non-uniform refinement doesn't have those two baked in, but it does have support for correcting them if the higher order aberrations and per particle defocus have already been estimated. So what you can do in non-uniform refinement, the easiest way is to do a regular non-uniform refinement and then use the standalone local and global CTF refinement jobs and then repeat non-uniform refinement again. So that'll get you one round of refining the structure, refining the CTFs, and then refining the structure again. You can repeat that again if you'd like. I would highly doubt that there are going to be major returns from doing so, but you can do that, and that's the only way to do it right now. You can also do a regular refinement with CTF refinement inside of it, and then use the output of that for non-uniform refinement. But in that case, you'll have, again, more trouble because you'll be using the output, or you'll be using the reference structure from regular refinement, which is definitely not going to be great because of the micelle and everything else that we just talked about for doing the CTF refinement. Once again, all these things are definitely worth questioning and wondering, but you should always just try CTF refinement. Same with non-uniform refinement. With non-uniform refinement, we've never seen a case where it makes things worse, only that it makes things better. Even on non-membrane proteins, lots of examples that we've seen. CTF refinement is not like that, so it's something that you just have to try.
Great. Two more questions on non-uniform refinement. Are there any parameters that we would want to adjust in non-uniform refinement if we were working with a membrane protein in detergent versus one in a nanodisc? Is that something that we can say right now, or is that also a matter of experimentation?
In my experience so far, I haven't found anything like that. It's not a question that I've actively explored, but I don't think you would expect there to be anything, mostly because the purpose of non-uniform refinement, and actually this makes it kind of bad for a practical tutorial, the purpose of it is to remove the necessity to tune any parameters or make any decisions about the protein or its regularization. So whether you have a nanodisc or a micelle, the algorithm itself will self-optimize its regularization scheme against the data through the cross-validation method that we used. And it will do something different in both cases by itself. Not something that you have to manually choose.
Great. Last question that we'll be able to get to right now. And I just want to say again, as I mentioned yesterday, we're doing our best to take all the questions. There are many, but it doesn't have to end today. So if there's something in particular that we haven't covered, or if there's something particular about the data that you're processing, the discussion forum, discuss.cryospark.com, is a really good place to post, because other users will also be able to help you out aside from our team. And you can always send us an email if it's something more sensitive at feedback@structura.bio. So last question on non-uniform refinement. From a theoretical perspective, what's the difference between the non-uniform refinement strategy that you explained versus applying a real space mask to exclude unnecessary background?
Great question. So in the case of a membrane protein in particular, there's a huge difference. If we think of what non-uniform refinement is doing, just go back a few slides to the figures here. The regularization that we're applying is specifically looking to correctly regularize different parts of the structure to account for their spatially varying properties, but it's not ignoring any part of the structure. Masking, which is different, first of all, because it's manual and you have to make the choice of which parts of the structure to include or exclude, is doing something very different in that it is breaking the model. It's saying that, you know what, there's something in the structure that I'm just going to pretend is not there when I'm aligning particles. And that's a very dangerous thing to do with a membrane protein, because the micelle itself, in terms of electron density mass, is very massive. For example, in this example, the micelle is a big fraction of the density of the actual thing that's in the ice. If you mask it out, now you're aligning a protein structure with no micelle to images that have something totally different in them, which is a protein structure with the micelle. And so it's very likely that the alignments will be totally wrong or get even worse because of that masking. So it's critical not to ignore or exclude something that's disordered. Unlike noise, which we want, like solvent, which we always want to ignore because what we're saying about it is that it has no density of significance, and we're trying to resolve density that is above the solvent density. Unlike that, disordered density, protein density, like if you think of an individual single particle image of a membrane protein with a micelle, the atoms in the micelle are just as real as the atoms in the protein in that single particle image. And they diffract electrons just as much as the protein. We can't ignore them, so we have to treat them differently because they're always in a different configuration from image to image, and that's what regularization is about. But we can't mask them out and just totally ignore them. Hopefully that answers the question. So in general, you can try doing masking of micelles, but it's unlikely you'll get very good results, especially in a case where the micelle is actually causing a problem because the density of the micelle will be so high, and when you ignore it, it's going to totally change what happens in the alignments.
Great, thank you. I think this is a good time for our 10-minute break, and perhaps we can resume at 3 p.m. Pacific time.
Excellent.
Okay, so 3 p.m. I'll set it up. Thank you.
Thanks.
Okay, great. So I'm actually going to take over for a bit. Hopefully it'll be interesting as the past 12 hours with Ellie. But I'm Stefan. I'm also on the CryoSPARC team, and hopefully everyone can see my screen.
Yes, we can. Thank you.
Awesome. Okay. So today I'm going to be briefly speaking about CryoSPARC's architecture, some hardware considerations, technical troubleshooting in CryoSPARC, and data management. So CryoSPARC at a high level can be split into two different modules, the master and the worker. The master is a single module that is made up of a few different services. First, the Python server we like to call command core. This API server schedules jobs and interacts with the MongoDB database, which holds all metadata in CryoSPARC. There's also the web server, which is built in React and Meteor and is what powers the CryoSPARC user interface Suhail presented yesterday. These services are quite lightweight and they can be run on a very modest workstation. The other half of CryoSPARC is the worker module, which is made up of Python and custom CUDA kernels that power the GPU accelerated jobs in CryoSPARC. A worker module is installed on each GPU workstation that is connected to a CryoSPARC instance. So given this master worker configuration, you can install CryoSPARC in a few different ways. The simplest is a single workstation, where the master and worker modules are installed on a single machine. The most flexible is, of course, the master worker setup, where the master is installed on one machine and it coordinates multiple GPU workers. For maximum scalability, you can integrate CryoSPARC with a cluster workload manager like Slurm, PBS, SGE. And here we'll see an illustration of a single workstation where both master and worker are installed on a single computer. And the only requirement is that there's NVIDIA GPUs on the machine. So the master worker setup is similar. The only additional requirement being that all nodes have access to a file system that is shared. The file system is where raw, intermediate, and final results will be written to. Having a shared file system is very important, as this is what allows the CryoSPARC user interface to show results that jobs have created. Note that the master instance doesn't need to be hosted on a separate instance. It can be hosted on one of the worker nodes as well. The cluster integration is very similar to the previous setup, where the master node submits jobs to the cluster workload manager like Slurm, and Slurm will take care of assigning resources required to run the jobs. The same bulk storage for the shared file system is required. Finally, the three main requirements for all setups is that 1, all nodes have access to a shared file system. 2, the master node can SSH into each worker node. This is actually how the command core module runs jobs on each GPU workstation. And third, all worker nodes can access the master node through a few predefined and configurable TCP ports, which enables communication via the RPC API. By default, it's 39,000, but you could change it to whatever you want. Okay, I'm gonna talk about a few hardware considerations, especially for CryoSPARC, and highlight characteristics that are relevant to cryo-EM data processing in CryoSPARC. So the first is the CPU, where a lower core count but higher clock rate is more beneficial than a larger core count at slower clock rates due to the way that the command core and compute libraries are written in Python. The next piece of hardware is very important. It's the SSD, which is used to cache particle images during reconstruction jobs. These jobs require random read access to the images, and having a fast storage medium, preferably at least 1 terabyte of a PCIe-based NVMe SSD, will greatly reduce runtimes. The network also has a similar concern. Since cryo-EM produces large amounts of data, you will most likely be running a raw storage server separately that is mounted onto each of your workstations via the network. Having a 10 gigabyte or more connection is very beneficial, especially during the pre-processing stages of processing, where large movies may need to be read from spinning drives and streamed over to the worker. Finally, the GPU is arguably the most interesting piece of hardware in cryo-EM data processing. All modern GPUs manufactured by NVIDIA will do the job perfectly fine, as long as they have at least 11 gigabytes of GPU memory. You may find that for your purposes, 11 gigabytes of GPU memory may not be sufficient for the size of volume you're trying to reconstruct. In that case, NVIDIA has 16, 32, and even 48 gigabyte GPUs available. So for more information, check out our guide, which has all everything that I'm talking about, including system requirements for CryoSPARC for both the master and the worker, including recommendations and even what we use at Structura to do benchmarking. Yeah, it has a lot of useful information. Okay. So now I'm going to switch over to technical troubleshooting in CryoSPARC. And to do this, I'm going to show an example of a job that has failed and the general method I would use to try and troubleshoot the issue. So I'm going to switch over to our production instance. And right off the bat, you could see that there is a failed job here in this project that pulled up, J17. So I'm just going to open this up. You can tell it's failed because it says failed and it's in red. And I'm going to look for red events in the stream log, they contain the tracebacks of the error itself. Note that the most recent call is last. So we'd start with the most recent event and then work our way up. So here I see the job failed because of an assertion error, not enough patches to constrain spline fit. And I actually intentionally made that error. I put an absurd number for the spline calculation. But if you, obviously you know what the error is because I'm telling you it, but if you couldn't tell based on these tracebacks, you could then move to the command line and use log commands. So I'm gonna switch over to my terminal here. And the command I'm gonna use is CryoSPARC-M, which is our management script. Job log and supply the project number and job number. So if I hit that, I'm going to see a bunch of information that came from the standard out of the job process itself. And I'll have a bunch more information that I can use to try to figure out what went wrong. If I still can't figure out what went wrong here, I can actually take all this information and move to our discussion forum that Sarah was talking about, search an error, and there should be stuff there. If I haven't found anything, I can always create a brand new topic with logs, the job type, maybe my system specs, everything you could provide that will make our lives easier to debug the job. Okay, great. So there's also a great resource on guide.cryospark.com that has all the logs that I've just talked about, well, the CryoSPARC-M job log, as well as other logs for the core services in CryoSPARC and how to use them. There's also a great troubleshooting guide that covers many different use cases and common errors in CryoSPARC and how to deal with them. Okay, now I'm going to go over some data management aspects in CryoSPARC. So CryoSPARC has several critical tools and features for dealing with cleaning up, archiving, transferring, exporting, and importing projects. We've also created tools for exporting and importing individual jobs and individual results of various types like particles, exposures, volumes, and more. So all these tools allow you to do things like sharing. So moving a project from one instance to another, taking a project home that started from a centralized facility, uploading your job to Empire. You can also save space with these tools. So we have the tool called Clear Intermediate Results, which allows you to clear intermediate results created by iterative jobs like 2D classification and refinement. You can also archive projects for later retrieval to also save space. And you can also do some really advanced workflows like editing a CryoSPARC file programmatically, a .cs file, I mean, and importing it back for some advanced workflow. So right now, I'm going to be mainly speaking about sharing jobs externally, but if you want to share your projects amongst users in the same instance, you could use the share with users option found in the project details panel. So what I mean is I'm going to move over to a production instance again. And if you see here, I'm going to click on a project, and on the right-hand side, if I own this project, I'll see the option share with users. And I'm going to click that, and I can actually give read and write access to every one of these users that exist in my instance. And that's only if I own this project. If I don't own this project, I won't have the option. I'm also an admin, and that allows me to see all these different projects. So that's if you want to share within your instance. Now, let's say if you want to share a project. One thing to note in CryoSPARC is that all projects are self-contained. This means if you want to share a project, there is no need to press an export button. All you need to do is copy the project or compress the project folder itself and share it with the other user. The other user can then put the file on or folder onto their system and go to the projects page and hit this import button right here that you see. You can then specify the actual directory to the project folder, and it'll be imported right into the instance. So jobs, on the other hand, are a bit different. For jobs, you will actually have to press an export button. And I'm going to just show you how to do that. So let's say if I want to export this extract from micrographs job, I would click on it, and the details panel will show up on the right. And then I'll have this option to export job. And if I hit yes, I'll get a notification saying, you know, this job is exporting. And once it's done, I'll get some text right here saying writing CS file and CSG file to a specific folder. So I could take that folder and then compress that folder or copy that folder to a different user. And that's how I would share that job. And why we need to do this is because in CryoSPARC, we usually have pass-through files. So we don't actually send data from one job to another. We use CS file, CryoSPARC files, that contain metadata about the actual job. So usually what you'll see in a job folder is symlinks to other jobs that contain the actual data. And when you hit export job, what it does is it consolidates all that data into a single folder. And that just makes it easier to actually move that folder from one instance to another. So similarly to projects, if you want to import a job, you would have to go into a workspace. So I'm going to go into P32, and I'm going to go into workspace 1, and then I'll see at the top, similar to import projects, import job. So I can hit import job and then specify the folder of the job that I've just copied onto my system, and then I'll have that job in my instance. So that's importing jobs and projects. Now, importing, let's say if I want to export particles or volumes or a specific result group, we also have a utility for that. And what you could do is, I'm going to open up this extract from micrographs job again, and you can go to the outputs tab. Now the outputs tab lists a couple groups here, micrographs and particles. And on the left side, you can see the export button. So this export button does the same thing that an export job button does, but for the specific result group itself. So if I hit export right now, I'll get a notification. And in the same place, I'll get another event text saying writing CSG file or CS file to the specific folder. And I can then take that folder, and it would only have the particles that came out of this job. And I can compress that or copy that and share that to another user. But the only difference now is that since I'm not importing a job or a project, we're going to use the import result group job to actually import the result group that is copied onto our system. And you would present the CSG file. So CSG file would be found in the actual folder. And what that is just a reference to all the different data inside the actual folder, like the CS file and the particles or anything else that has been brought along. And so another common use case, and we get a lot of questions, is how do I export particles to use in a different program? And for this case, you could use the PyEM package created by Daniel Lassernow. So I'm going to show his GitHub page right now, but it's a great utility. It's called CSpark to star. And what it does is it takes a cryospark.cs file, converts it to a .star file so that you can actually use it in a different program. And you can look at his wiki, and it has a bunch of information on how to install it, how to actually use it. And if you go onto our discussion forum, and you'll see a lot of support topics that cover a bunch of different use cases and errors that have come up over time. And again, I would like to mention that everything that I'm saying here is available on our website, in our docs, including a bunch of different use cases, and you can read all about that on our website. So yeah, this concludes my presentation. Thank you. So I'll hand it over to Sarah for some questions.
Awesome, thanks, Stefan. We do have a few from across the board. So first off, in terms of actually installing CryoSPARC, you did touch on this, but the question, if we can just recap, is it possible to install CryoSPARC on an HPC node?
Yeah, so I'm assuming by HPC node, maybe you mean like a cluster. And so you have this big cluster that your university or institution has, and you want to, download CryoSPARC for yourself. This may require actually talking to the cluster sysadmin. But if that's not the case, you can actually, usually what would happen is you install CryoSPARC on an edge node that has access to the command line tools for the actual workload manager. And that, what it would do is you just basically supply a cluster info.json. So it's a JSON file that basically tells CryoSPARC, these are the different commands that I would need to execute in order to run a job, delete a job, get info about a job. And then once you tell that to CryoSPARC, you can queue jobs automatically, like normally as you normally would, and then that would submit a job to the actual cluster. I think that's what they meant by HPC.
Yes,
but
I think
yeah.
so.
Okay.
Thanks. Awesome. Speaking of actually being able to continue on with an existing installation, is it relatively straightforward to add a worker node to an existing installation?
Yeah, of course.
We have we add a microscope and a GPU node specifically just to do on-the-fly processing for the microscope, but we already have an existing CryoSPARC installation.
Yeah, yeah. So actually, this is sort of what the single workstation setup is. That's what it came from. The simplest setup is to obviously just install CryoSPARC master and worker on a node that has a GPU, but you're going to eventually scale up. And once you add a GPU worker, you can go ahead and just connect a worker node. We have a simple utility called CryoSPARC M Connect, and it will do the work of detecting all the GPUs on the system, and it will create a separate lane for you, and you can now queue jobs to that lane. And you can always, you can mix and match. You can have another single workstation. Like let's say if you keep a workstation and just add a GPU workstation, or you can add a cluster integration itself. And maybe you have like a local GPU workstation and you have this huge cluster that you only use, very rarely. So you can mix and match.
Awesome. Can you speak a little bit to being able to access CryoSPARC remotely where it's actually installed somewhere else? And the specific question actually came from one of the practical participants in this workshop. So they say, have I been processing data on my computer, on the Stanford Slack server, or on your CryoSPARC server? And they're accessing all of this remotely.
Yeah, for sure. So for the practical participants, you've actually been processing data on the Slack servers. And what happens is essentially they're running the service on their machines, and they're routing that port that CryoSPARC runs off of through a service called Nginx. And you just go to a website and it basically translates you into that server. We have a whole guide for this on our actual website, on our new guide. And when you start CryoSPARC, you'll get a message that says, from this machine, you can access CryoSPARC at localhost at 42,000, because CryoSPARC is hosted at a specific port, the web app. But if you're on the same network and that machine has a host name, you can actually go to that host name at that port. And like I mentioned, Nginx, it's a utility, it's a reverse proxy. But yeah, you can essentially map an actual host name to a port, and that'll direct traffic to that port. So yeah, that's how you've been doing it. But yeah, everyone on the practicals have been working on the Slack servers.
Awesome. Can I run CryoSPARC on a MacBook Pro that has a GPU?
Interesting. If you have a MacBook Pro with a GPU that works with CryoSPARC, it might be old, because we only support NVIDIA GPUs. And lately, Apple dropped support for AMD, for NVIDIA GPUs. They only have AMD GPUs. So if you have a fairly old MacBook, yes, technically you can. Although you might find with later versions of CryoSPARC, the support is a bit limited because we have developed things that are specific to Unix or Linux systems. So yeah, might be hard, but it's possible.
So we have a lot of instances where we obviously are not storing all of our terabytes and terabytes of raw movie data on the same place where we're actually processing all that data. So can you explain more about how we can read raw image data remotely from the shared file system to our workstations without actually transferring all of those large files over?
Yeah, definitely. So like I was saying with the master worker setup, there's a requirement that there should be a shared file storage that is accessible across all nodes. And taking off this, what you should do in your case is you should create a mount point on your local system and use NFS, so network file sharing. And essentially what that'll do is it will allow you to host that raw storage server that's probably huge at a folder location on your local system. And that'll allow you to directly access that raw storage folder. There are a lot of tools like that, and a lot of guides available actually on how to do this. We actually do this at Structura, but that's the most flexible and easiest way to do it. And it allows you to actually, you probably will have to, well, right now you're probably copying data from that raw storage to a specific node that you want to process on, but with this system, you can process on any different workstation because the actual mount point is available on all instances. So that'll make it definitely easier, and it's possible.
Awesome, thank you. And finally, we talked a little bit about the cluster setup for physical clusters. Can you talk a little bit about running CryoSPARC on AWS?
Yeah, actually, we have a lot of instances where AWS has been used, specifically parallel cluster. So parallel cluster is essentially a managed cluster system that will allow you to select one of their AWS nodes to use as a worker, and you select a master node, and it works the exact same way. You would install the master node on that edge node, and then you would basically use Slurm, PBS, or I think it's Slurm, which would be the most reliable to use on AWS parallel cluster. And you would integrate it as normal. You would specify the commands to CryoSPARC, and then CryoSPARC would go ahead and submit jobs to the Slurm cluster. And all the management is done on AWS itself. You can even do this without parallel cluster. You can just do this with EC2 nodes. And I guess a single workstation would be the easiest, where you just provision a node, maybe something that has obviously NVIDIA GPUs, install CryoSPARC on it, both the master and the worker, and you can use it like normal. So yeah.
Awesome. And actually, last question, this is more to do with hardware, specifically GPUs. It's often that one bumps into an out of memory error if you have a system with 2 GPUs, each with only 8 GB of GPU memory. If you do have this kind of setup, what can you do to work around, or when might you encounter a memory error with 8 GB of GPU RAM?
So 8 GB of GPU RAM, definitely on the low end. We would recommend 11 GB for sure. And that means, I guess it would be the 2080 Ti's or the 1080 Ti's. But if you do come into that error, we do have a lot of developments coming out right now from some scientific developers on our team, Harris, who's been working on optimizing our motion correction job, which previously had run into errors on 8 GB cards. And so now with the patch, actually on 2.15, there should be no problem using 8 GB cards for patch motion jobs. Refinement, on the other hand, should be completely fine. I believe 11 GB, you can process 1024 pixel box size. So it's pretty large. You probably aren't. Most comments like around 256 to 500. But yeah, so maybe update to the latest version and you should have a better time.
Awesome. Okay, awesome. Thank you so much, Stefan. That was really informative.
Great. Thank you.
Yeah, that was great. Thank you, Stefan. All right. Thanks, Stefan, again for explaining exactly how CryoSPARC works and how people can actually use it. I believe what's going to happen next is that I will continue in terms of the practical and slides into the next set of topics. So let me just start sharing my screen. Make sure they're at the right place. moment. And now I will share my screen. So two. All right. Can everyone see my screen?
Yep.
Yep. Great. And is the session still recording?
Yeah, it looks like it.
Great. Okay. Where we were last is that we had been working on talking about non-uniform refinement and how it works. We had started processing a GPCR structure. I just want to let everyone know, the practical participants, that probably your GPCR refinements are done at this point, and you can go ahead and inspect those just as a reminder. I'm not going to take questions right now, but what we're going to start talking about next is an exciting area of new developments in single particle EM methodology, especially on the compute side, and projects that we've been working on for quite some time, to start to look at the problem of continuous heterogeneity and conformational landscapes of protein molecules that don't appear in only a single or multiple discrete conformations. As an example, we've seen this protein now a few times, the NAV 1.7 channel. It is, as we mentioned, a therapeutically relevant pain receptor, and it has an open and closed state. As an ion channel, it has both open and closed states, which are easy to distinguish because they come in forms that are discrete. So particles are either in one form or the other and not somewhere in between, generally speaking, and they have a clear conformational difference. The active state has the pore open and the structure is fully symmetric. And the pore is sort of a square shape made of transmembrane helices that create a square pattern. The deactivated state, on the other hand, the pore is closed, and the membrane protein actually adopts a different symmetry. Now, instead of fourfold symmetry, only twofold symmetry, and the pore becomes elongated and rectangular and closed. So those two discrete states are clearly separatable from the image data because the two different structures are so different from one another. They also form a discrete landscape rather than a continuous flexibility or a continuous conformational sampling. So those separate states can be resolved by tools that we've already discussed. Those were ab-initio reconstruction, heterogeneous refinement, and so on, to classify different discrete structures. What we want to think about next, and this is something that I mentioned earlier, is that this protein, along with many others, shows clear signs when we resolve its density that there are parts of the structure that are flexible, where the protein itself exists in a family of configurations that are not discrete, but a protein can take on any position along a continuous flexibility and may appear in any of those positions rather than just one or another. And because of that, the recovery of 3D structural information is severely limited in certain parts of the structure because those parts are flexible. And all of our mathematical models, all of our reconstruction algorithms so far have assumed that the structure is rigid, and that in every single particle image, we're seeing the same object in the same way, in the same shape, from a different direction. So what we need in order to deal with this scenario is a new way of thinking. We've seen this a similar notion of sort of conformational spaces in the discrete case. For example, this ATPase molecule that we earlier saw when we were talking about multi-class heterogeneity. This is a very interesting molecule, and there have been several follow-up studies by John Rubenstein's group here in Toronto, who's been working on ATPase molecules for quite some time. They've gotten some amazing, beautiful, very high-resolution structures of all kinds of different ATPases. The data that we're actually going to be using today for the practical comes from a 2016 paper. It's 200 kV data, so a little bit collected on slightly older hardware, but we're not looking for high-resolution structures right now. We're really looking at the dynamics of the molecule, which are fully visible in this data set. So we already saw that in this case, we were able to, with ab-initio reconstruction or heterogeneous refinement, separate the overall set of particles into 3 configurations that are all in different rotary positions of the central stalk. And so those different rotary positions are discrete conformational changes. The central stalk rotates between them in a continuous sense, but in terms of the energy landscape, we're extremely likely to find a particle in one of the 3 positions and not in between, at least when the protein is inactive and it's not actively rotating. So because of that, those different conformations are discrete conformations, and we can separate them easily, and each one we can treat, once we have found the particles, as a homogeneous, rigid object. There's just 3 variants of it. That's not the model that we want to go forward with, though. And it turns out even on this data set, there is lots of interesting flexibility that happens in the molecule that we can now resolve. Zooming out a little bit and thinking about conformational heterogeneity at a high level. From a conceptual point of view, what we're trying to do in the case of conformational heterogeneity is consider that the particles that we capture in the image, in the microscope, the images that we capture, sample, each individual image is a sample. So collectively, they together sample the space of different conformations that the particle can take on. If that's a discrete space, then we will have clusters of images representing each conformation. And if it's a continuous space, like the molecule has flexibility, then we'll see particles that sample that whole range, ideally. So if we think in a sort of conceptual sense of a space, the sort of mathematical notion of a space, of all the possible particle images that could be generated, clustering of those particles that we observe into different categories in the space is what amounts to 3D classification. When our model says that we have multiple different discrete objects in the sample, each of those discrete objects will give rise to images that are similar and cluster together in the space of all particles. On the other hand, if we had continuous flexibility, instead of discrete clusters, we might find a continuous band, or what we call manifold, of particles in that space of images. So that's the kind of large overview picture of how we can think about conformational landscapes. The existing algorithms that exist today, 3D classification or heterogeneous refinement being a good example, can already resolve significant biological insight due to the fact that many particles do have discrete heterogeneity. But there are some questions that arise. Discrete heterogeneity, like I said, corresponds to clustering, finding bunches of particles that are all the same in terms of the conformation they represent and separating them. That's clustering. But there are some questions that arise. How do you know how many classes there are? In real life, we can't actually see this space of all particles. Like mathematically, the space of all particles is a gigantic, extremely high dimensional space. The number of dimensions is equal to the number of pixels in every image, since that's how many different ways an image could be different from one another. And we can't actually visually look at that space and see clusters. We have to use a clustering algorithm like heterogeneous refinement or Ab-initio reconstruction, and then look at the clusters that it finds and say, did we find all the clusters? Could there have been another cluster that we missed? Could these clusters be split into subclusters? We can't actually see that or know that ahead of time. So there's a big question of how many classes really are there? How many clusters should we split? And the bigger question, how do we deal with continuous heterogeneity in this framework? Continuous heterogeneity won't cause images to form clusters. It'll cause images to lie on a continuous manifold. That's the space in which we're operating, and what we've been working on and what we've done to develop 3D variability analysis, which is what we're going to talk about next, is to come up with a new algorithm with a different model in a world that many people have been working in. There's lots of literature and lots of work in trying to recover continuous manifolds of 3D structures from cryo-EM data. It's a dream and a goal for single particle EM as a field to be able to look at and investigate from an experimental perspective protein dynamics. And it's totally conceptually possible, computationally very difficult, and algorithmically very difficult. And 3D variability analysis is a step forward in that direction. So before we get going to explaining the algorithm and how it works, and I have a bunch of slides for that, we're going to just jump over to our CryoSPARC instance and start a little more practical data processing. So first of all, in our projects for all the practical participants, we're now following the instructions for the ATPase molecule. If you can please create a new workspace. It should be probably second nature at this point. In that workspace, create a job, again, import result groups, and specify a path to a CSG file, again, looking for inside the Scratch training projects folder, your own project number, which is in the top left corner if you're in CryoSPARC, so in my case, project 6. And inside of that project, you will find the Slack workshop data folder. Open that and look for dataset number 7, ATPase. There's also dataset number 4, ATPase. That folder is accidentally empty. Dataset number 7 is the one we're using. And look inside the particles folder and you will find a CSG file. So select that, and that's going to import all of our particles that we need. And at the same time, create another job, this time an import volumes job, 3D volumes. And with that job, choose the volume data path, again, inside of that same 7_ATPase folder. It's called mask.mrc. And that's a volume that contains a mask that we're going to be using during 3D variability analysis. And when you're importing that volume, be sure that when to change the type of volume that you're importing from map to mask. The import volumes job in CryoSPARC doesn't know what it is that you're importing, and every type of volume is stored as an MRC file. And so you have to tell it that this is a mask that we want to import. So you can run that job. And then you should have two completed jobs, one with an imported mask and one with particles. Next, we're finally going to create an import 3D variability analysis job, which we can find in the builder, 3D variability. That job is going to take as input our imported particles and our imported mass. And what you'll notice if you expand the dropdown for the particles that we just connected, 3D variability is actually going to be using three different things. One are the blobs, which are the raw particle data. One is the CTFs. And the last one is the alignments. This is something I'll come back to, but in 3D variability analysis, we assume that we already have the particles aligned to a consensus refinement, meaning that in order to use 3D variability analysis, what you should always do is take all of your particles, whatever conformation or class they're in, put them all together and do one reconstruction using a refinement job like homogeneous refinement that gives us a consensus structure. So that might be an average of all the different classes and alignments for every particle. And we know that those alignments might be slightly wrong because we haven't dealt with the conformational differences, but that's not important. 3D variability will actually help us figure out that problem. So those are the inputs that we need for 3D variability analysis. Now I'm going to explain what all the parameters mean, but right now we're just going to set the filter resolution to 8 angstroms and go ahead and queue that job. I'm not actually going to do that on this server since everyone else needs to use it, but I'll come back to my own instance where I have already run the 3D variability analysis job. So that's all we need to do right now, and I'll just jump back to the slides and continue. I'll just give another minute for practical participants to follow the instructions.
One question on this note. How do we actually create a mask for this 3D variability job? The practical participants are working with partially pre-processed result groups. So if you can just mention that.
Creating a mask is a bit of a, I wouldn't say art, but it's a skill that you have to learn. It takes a few steps because usually if it's a mask that is in any way interesting and that you've selected a region of protein, you have to manually make that selection. For example, a common way to do it in UCSF Chimera, which is a very great program, is to manually erase parts of the structure and use the resulting part of the volume to create a mask. Unfortunately, we won't have time to go into the details of exactly how that's done today. But in CryoSPARC, we also have several tools that help you make a mask once you've already imported the relevant density that you maybe have created in Chimera by manually selecting regions. And those tools are called volume tools. Volume tools job allows you to connect a volume or a mask and then do a bunch of operations, whether that's resampling, cropping, low pass filtering, flipping. You can take in a volume and output a mask. And when you do that, you have choices to, for example, dilate the mask, threshold the volume, pad the mask with a soft edge, invert the mask, and so on. So a lot of those operations are available, but the actual act of selecting parts of a structure has to be done in a 3D visualization program, right, like Chimera. And then you can use the import facilities to import volumes or masks from those sources. Okay, so now that the practical participants have been able to construct that workflow and it's hopefully running, I'm going to start talking about 3D variability analysis. This is another topic that we've recently had time to write a paper about. We originally released 3D variability analysis in CryoSPARC in June, I believe, last year. And it's been widely used as well. It was also used in recent coronavirus spike protein structures to resolve flexibility of the receptor binding domain, which was really exciting and hopefully will be useful in further studies. The actual algorithm itself is described in a lot of detail, as well as a lot of the prior work that has come in this field. Lots of different people have been working in the field of continuous flexibility, and it's very much a difficult and open problem still. 3D variability is only a first step, and it does work very well in practice. As far as we know, the first method that's able to resolve high resolution motion in multiple different modes, different types of motion at the same time, and at high resolution. And it's also the first method that can work for smaller proteins, not just very large complexes like ribosomes and spliceosomes. It works for membrane proteins and small proteins as well. I'm going to talk about exactly how it works and how to interpret the results. It's something that takes a little bit of thinking to make sure we understand collectively what it is that 3D variability does, what it models, what it doesn't model, what its limitations are. But overall, the purpose of it is to directly determine and resolve 3D motion and continuous or discrete conformational changes from a data set of particles without some of the pitfalls that we just discussed about clustering methods. The model behind 3D variability analysis is very different from what we use in any kind of clustering, whether that's ab initio reconstruction or heterogeneous refinement or 3D classification of any other kind. 3D variability analysis does something fundamentally different and models the 3D structure very differently than we've seen before. And that's exactly what gives it power to resolve things that we couldn't previously resolve. So I'd highly recommend anyone who's really using it to read the paper. We tried to make the paper a little bit long and go into a lot of conceptual detail and make sure that it's easy to interpret the results and understand what the outputs mean. This page has a little bit of math, but I'm going to slowly go through it and try to give a sense for what are the concepts and objects that we talk about in 3D variability analysis, since they are very different from so far what we've seen. We're no longer just dealing with electron density maps and particles and alignments. There are now some extra objects. So first of all, as I said, the purpose of 3D variability analysis is to directly determine 3D motion and conformational chains. If we think back, and I'm not sure if you remember yesterday, we presented a sort of diagrammatic view of our inference problem from a computational statistical perspective. What we're trying to do in single particle EM is that we observe a bunch of images, X. We get some microscope settings about those that we know, and those can be called theta or C for the CTF. We can assume that we know those. There's a pose that in general is unknown, but for the sake of 3D variability analysis, we are going to assume that it is known. So like I said, we're going to start with a consensus refinement and take the poses from that refinement and fix those. And finally, there's the 3D structure itself, which previously in the models that we've considered is represented by one or more discrete static 3D electron density maps. So V is literally, in the homogeneous refinement case, a single electron density map, or in heterogeneous refinement or ab initio reconstruction, it's one of K discrete, maybe that's 3 or 5, whatever the number of classes was, discrete different 3D electron density maps, and they're not related to each other in any way. Like other than the particle images, there's nothing that forces heterogeneous refinement to recover multiple structures that look similar in any way. It's only the data that says that. So the standard model, thinking of just, let's say, homogeneous refinement, if we think of the equation that defines what is an experimental image, we would say that an experimental image can be modeled as taking the single volume that we may or may not know, projecting it with some projection operator based on the angle that we're looking at the structure. I'm sorry, I use different letters here. Phi represents that angle, the pose, and then corrupting it by the CTF for that particular image, and then adding noise. That's the standard formation model for single particle EM. In 3D variability analysis, what we do is extend that model in a different way. Instead of saying that there's just one or more static structures, we say instead that at the fundamental level, a single particle image is generated in the following way. First of all, we have a new object, this cursive V. That thing is, it represents a family of different 3D structures, electron density maps that are all related somehow, a family of different structures that covers the conformational landscape of the molecule. So for example, in a simple case, imagine we had a protein that has 2 subunits, an overall complex, and the different ways in which it can appear are either both subunits or one subunit or the other subunit. In that case, we could think of that scenario as 3 different classes, like the intact complex, subunit A and subunit B separately, or we could think of them as all being related, and they're part of the same family, and the family is a is a structure that's made of A and B parts, which are independent, and the A and B composite part, which is another member of the family. But it's not a separate volume. It's actually connected to the A and B parts because it's made of both of them together. Similarly, if we had a flexible molecule, we could think of every different flexed version of the molecule as a different electron density map that we have to model. Or we could say, no, actually, our model is much simpler than that. We have one electron density map and some motion. And that motion defines a family. So the notion of a family of structures captures every different kind of heterogeneity that we want. How we actually implement and represent a family is what gives a model that we're going to use, for example, in 3D variability analysis, a certain amount of power and a certain amount of practical usefulness. The important thing about a family of volumes is that it's not a volume, but it can generate a volume if you provide it with what we call a per particle latent coordinate. That coordinate is one or more numbers that describe for a single particle image which member of the family is in that image. So if it's a flexible molecule, the family is actually composed of the structure and the motion, but the per particle coordinate tells us for a single image, whether it's on the left by 10 degrees or on the right by 5 degrees, that is the per particle coordinate that we are going to talk about. And that's a very important concept. So the per particle coordinates are now a new object that we haven't had before, and we don't know those coordinates. We also don't actually know the family of volumes. That's exactly what we're trying to find out when we're solving a heterogeneous problem. Other than that, the model is more or less the same. We project the structure that comes out of the family after picking the right member based on the coordinate Z. We project that based on some poses that, again, we assume we know, corrupt by the CTF, and then we can scale that by some unknown scale factor. This is not too important, so I'm not going to mention it anymore, but alpha is just a contrast level, since every particle image may have a different amount of contrast due to the ice thickness. So that's also going to be incorporated into the model. So now this is what we're saying a single particle image is. One member of a family indexed by Z, projected, corrupted by the CTF, creating the image with noise. To get specific with 3D variability analysis, we're going to pick a very particular family, type of family, and that's a linear subspace model. It's the simplest family we can think of other than a static structure. The beauty of it is that with this family, it turns out, and the main contribution, the main new thing in 3D variability analysis is that we have an algorithm to actually solve for the parameters and the density that makes up this family. Because it's so simple, it's possible. That family that we're going to think of is a linear subspace model. And that model is composed of the following parts. There's V0, which is a static electron density map, the consensus refinement. So V0 is just the structure that we would have got by refining all the particles together in a standard model. That's what V0 is, the basic regular refinement structure, electron density map. But the family says that we can generate any other member of the family by taking the basic structure and adding to it some amount that's captured by Z of K different variability components. So each of these K different Vs, V1, V2, V3, are now, instead of being different classes, are different modifications that we're allowed to make to the basic structure in varying degrees, and the degree of modification is indexed by Z. So a Z value of 0 means we don't add any of that particular V, but if a Z value is 5, then we're adding 5 times that particular V to the basic structure. So the variability components are types of change that we're allowed to make to the basic structure. And there could be, if K is, let's say, 5, then there might be 5 different types of modifications we're allowed to make. And the Zs tell us for any given image, particle image, how much of each modification is present in the image. Hopefully that made some sense. And what we're trying to do here, what the family is trying to represent in a simple way, is this notion of a continuous manifold of 3D structures that could be present in the image data, instead of just a static structure. And instead of just a small number of discrete structures, we can now represent a continuous family. It's a simple continuous family, but we can take a basic structure and add some real value, some floating point number times a variability component to generate a slightly modified or more modified or slightly less modified version of that structure along 5 different, if K is 5 different dimensions. So hopefully that's sunk in a little bit. I'm not going to talk about the orthogonality constraint, but basically we're also assuming that the variability components are all different from each other. Overall, what this model does is it allows us to model a continuous family of 3D structures that fit the data. The hard part, of course, is after we've written down this model, which has been written down many times before, and it's kind of a straightforward, obvious choice for those who work in this field. The hard part is how do we actually estimate and infer what are the VKs? We know how to infer V0 because that's just how we do regular refinement. But now how do we infer what the VKs are? How do we figure out what are the different ways in which the molecule can change? At the end of the day, that's what captures heterogeneity. So what we've done in 3D variability analysis is solve that problem, find a way to optimize and fit and infer those variability components with a high degree of accuracy at high resolution with lots of particles in a computationally efficient way using full CTF correction and all the different poses that we could have. And what that ends up giving us is a method that can, I'll just come back to that, is a method that can resolve continuous heterogeneity and flexible molecular motion. But just to make the concept of a variability component and this whole subspace model more concrete and try to understand what can it possibly represent, let's look at a very, very simple example on the left side. This is an example of a reconstruction of a ribosome protein, large protein, easy to reconstruct. If we do a standard homogeneous refinement, we get a basic 3D density structure. And we've seen these plots in CryoSPARC before. There's our ribosome. Using the 3D variability analysis algorithm, on top of this basic structure, we can now estimate a variability component. Now, what exactly is a variability component? It is literally a 3D volume of values. They're no longer electron density values. They're instead differential electron density values. They're telling us how to change the electron density in the basic structure to form any other version of the ribosome that exists in the sample. So that's what a variability component is. If we take the basic 3D structure, and by the way, the variability component here is colored red and blue for positive and negative values. So we can have negative and positive values since we may want to remove density from one place and add density in another place in the variability component. So if we take the basic 3D density and add the variability component weighted by some amount, we can generate every other version of the ribosome that this model can model in the data set. And in fact, when we do that, and when we apply 3D variability analysis, we not only can solve the variability components, at the same time we solve, and we have to solve, what are the Zs for every image. Just like with 3D refinement, we were really solving the 3D structure, but we also ended up estimating the pose for every image, and we can make those diagrams of pose distribution. In the same way, we can make diagrams of distribution over per particle latent coordinates. And if we think of that for this simple ribosome example, there are lots of different values that this latent coordinate can take on, and we can make a histogram across all the particles of, for each particle, where did it fall on that spectrum? So some particles are on the negative side of the spectrum, some particles are on the positive side, and we'll see shortly that in many cases, the structure of that distribution over per particle latent coordinates actually shows us a whole lot of information about the heterogeneity present in the molecule. Okay, so I think that was a lot to take in, but one more important topic about the 3D variability model in terms of what it can capture. One of the things we're really after is modeling continuous flexibility. And continuous flexibility means motion. Parts of the molecule are going to move from one place to another. Motion isn't something mathematically that exactly fits into a linear subspace model. Motion, generally speaking, is not linear. But for certain kinds of motion and in certain contexts, the linear subspace model can actually be a very good approximation to molecular motion. As a very simple example, we've made some synthetic examples here. Consider a 2D version of the problem where now we're looking for motion of this black dot. This is some 2D electron density. It moves from one position to another in the image as we move from left to right in the top row. This top row are, for example, the experimental images we might observe in the microscope. The dot moves from one place to another. If we take those experimental images, put them into 3D variability analysis and solve for one 3D variability component under this linear model, what we end up with is this blue and red variability component. So it's telling us that in order to generate all of these moved dots at different positions, we should start with the average of all the dots and add some amount of this positive and negative shape. And if we do that, what we get is right here in the bottom row, from left to right, generated versions of the actual data under the linear model. And what we see is that it's a pretty good representation of the motion of the dot, especially if you consider thresholding this density at a certain level. Like, for example, when we visualize molecules in Chimera, we threshold the density at a certain level. Then we would definitely see the dot moving exactly the same way as in the original data. So in this case, we're seeing a motion that is about the same size as the size of the object or the resolution of the object. I mean, this dot maybe has lots of little tiny details, but maybe we just haven't seen it at that resolution yet. So this is a fundamental notion. We can resolve motion of objects at a length scale similar to the resolution and size of the object. On the other hand, if we had an object that's very small compared to the amount of motion that's happening, so now we have this tiny dot moving the same distance as the large dot, and we do the same thing, now we see that our linear model is actually much worse, and it doesn't really faithfully capture the motion of the dot. It smears out the dot along that motion direction. So that's a fundamental thing about the 3D variability analysis model, is that it can perfectly well model motion, but under these constraints that we should be looking at motion of a length scale similar to the size of the object that's moving. What that means in practice is that if we have a protein structure that has a huge amount of motion, we can only use 3D variability analysis to model that motion if we look at the protein at a very low resolution. Because at a low resolution, the protein is a big object with no small features, and we can see it moving by a huge amount. If we want to see high resolution motion, then we can see that for proteins that we can resolve to a high resolution and they have small amounts of motion at that high resolution. And we'll see this naturally in the results that we get. But this is what 3D variability analysis does and how it represents motion and what it can do for us. Okay. So,
Ali,
yeah.
I have a quick question. How is this different from, say, taking an object, which is the average of all the structures, and then treating each of the individual instances and performing a difference map?
Great question. It is very different because in this case, I mean, in the simple toy example that I showed here, there's no noise, there's no CTF, and there's no notion of 2D and 3D projection. So in the simple two-dimensional case that I'm showing here, what you're saying is right, that we could take the average of these structures and we could also compute, for example, their variance or a difference map or something, and it would look very similar to this linear model. And in fact, mathematically, they may even be equivalent. But in the real context of the 3D reconstructions that we're doing, those notions can't work and can't be applied, although many have tried to make variants of ideas like that to work in 3D. The reason they don't work, there's three reasons. One, that every 2D image that we collect in the microscope and every single particle image is only a partial observation of the entire 3D structure, and it's also only a partial observation of each variability component. For example, if we have motion of a subunit that in a particular single particle image is up and down along the electron beam, we won't see any change in the particle image. Whereas if we're looking at the same motion on the side, we may see a difference in the image. That's one reason. The second reason is that every single image is corrupt by a contrast transfer function. So unlike in this toy example, we don't ever actually see the object moving. We see the refraction pattern of electrons moving through the object as the object is moving, or with the object in different positions along its motion. Because of that CTF, we can't actually treat the observations in the single particle images as direct observations of the density or of the variability components. We have to actually account for the CTF when we're performing inference of the variability components, and that's not in this toy model. And lastly, the dimensionality of the problem is very different. In this toy model, we may have 1,000 pixels or something in total in the image. In 3D structures, we may have millions of voxels in our representation of the density. And so the size and computational ability to represent these objects or other statistics about them becomes impossible in very large dimensional spaces. Hopefully that answers the question. All that, what I just said, is in very much a lot of detail in the paper. So if anybody wants to read it, it's hopefully a good explanation. So what I'll do next is I'll go into the results, some of the results, so that we can get an idea of what these 3D variability components are, what they model, and what kind of motion they can resolve. Unless you want to.
We have a few kind of more theory questions that might be better to address now, just so we all have a fundamental understanding. So first of all, I'm not going to be able to go in a very particular order here because there are a lot of questions, but specifically with respect to the Z term, the per particle coordinates, what number or values are we actually using for those?
That's a good question. The values do actually have significance, but they're not as easily interpretable as, for example, a pose angle or something like that. They correspond to the amount of each variability component that we need to add to the basic density to generate a particular member of the family. The way that their units are created is that we actually enforce that the variability components themselves have a unit norm, meaning that each of the variability components has a certain amount of difference to the basic density encoded in it. And then if you multiply that by 1 or 10 or 5, you get a different amount of change to the basic object. Now the actual units depend on the data set, depend on the signal to noise ratios and the contrast and the overall units of like how many electrons there were and so on. So they're not something that you can interpret from one data to the next, not yet anyway, although theoretically that is possible and something that we can derive the math for. Right now though, you have to think of them as more or less unitless relative values between particles in the same data set. So a particle that has a -100 on one Z value and another particle that has a +100 are as different from each other as two particles that have 0 and 200 in their variability components.
Just to orient ourselves a little bit within the actual data processing workflow, you mentioned that we do need a consensus refinement before we can do 3D variability analysis. Is there also any minimum number of particles for us to know that our 3D variability analysis is reliable?
That's a great question. As a rule of thumb, 3D variability analysis does require a lot of particles, more than you would need for, well, to get good results out of 3D variability analysis, you have to have a great sampling of the conformational space. So naturally it takes more particles. And if you want to estimate more variability components, like with higher numbers of K, then you also need more particles. For exactly how many particles you need, I can't exactly say. It depends on many, many factors, but it seems to be that you can have particle sets that are sufficient to generate a single static structure, even at a moderate resolution, but not sufficient to be able to resolve 3D variability components. Typically, I'm thinking like you need hundreds of thousands or more particles most of the time to get good results, although the example data set that we're going to work with today has about 40,000 particles, and it's good for resolving 3 variability components. That's something that we need to explore a little more, including the validation of 3D variability components. And well, validation is actually, I mean, if we read the paper, there's some information about that there, but knowing which variability components are significant relative to each other is something that we need to investigate a little more.
And in the interest of keeping as many particles as we can, is it therefore better not to run heterogeneous refinement before 3D variability? And I guess intuitively, in a sense, if we separate out our particles into different classes, we sort of defeat the objective of doing this conformational exploration.
Yes and no. Yes, if the purpose of running 3D variability analysis is to resolve conformational differences that we could already resolve using heterogeneous refinement. If that's the case, then if we're double checking, for example, we should take all the particles that we have in total, put them into a 3D variability analysis, and we'll find variability components that show us the difference between different conformational states. And I'll show examples of that. But the more interesting version of the problem is where we've already separated the data set into as many conformationally discrete states as we can. And now within each of those clusters, we want to see what is the continuous flexible variability that is remaining after we've separated the data set into discrete clusters.
When we're actually setting up the job, is the number of variability components something that's determined without any input from the user?
No, that's something that user chooses. And in the paper, you'll see there's some interesting math behind why 3D variability analysis has a special property, which is that it always solves variability components in their order of importance. So for example, if you solve 3 variability components, those 3 will be the 3 most important in terms of how much variability in the data set they explain. And if you solve it with 6 variability components, you'll get the same first 3 and 3 more in their order of importance. That's an important property, and that's helpful because it makes it easier that you don't really have to guess how many there are. You can just solve as many as possible and you'll always get the most important ones present. The other mathematical property that's very interesting is that 3D variability analysis, unlike ab-initio reconstruction, refinement, and so on, doesn't have a problem of initial model bias. In fact, you can prove that it will converge and under some conditions, no matter how you initialize it. And so in practice, we actually initialize 3D variability totally randomly. And each time you run it, you will find within numerical accuracy and other randomness, the same variability components over and over. So you don't have to worry about, it's not like choosing the number of classes where you may be missing some or not. You can always just solve more variability components until you start to find that they are no longer capturing significant motion or variability or anything that you can interpret. Obviously, there's a limit in terms of like the signal to noise ratio in the data and how many variability components you can actually get, but you won't lose the first few if you just ask for more.
And now just to understand how 3D variability differs a little bit from other refinement and similar conformational heterogeneity exploratory approaches. So the first is, would the continuous 3D variability algorithm produce similar results as a multi-body refinement and other softwares like rely on, for example?
That's a great question. Yes and no. The multi-body refinement method is an extension of local refinement, which we'll talk about a little bit later. And both of those methods revolve around a model that is very different from this one. That model says that we are going to say that our protein is made up of a small number, basically 2 or 3, maybe 4, individually rigid components. And the only flexibility we're allowed is rotation and translation in a rigid body sense between the different rigid components that we've manually enforced. So in multi-body refinement, for example, you create masks to cut out regions of your protein and say, okay, now my model says, instead of 1 object, I have 3 pieces that can rotate with respect to each other. But each of those pieces is still assumed to be perfectly rigid. So it's not really continuous flexibility that's being modeled in multi-body refinement. It's a different version, a different simplification of the real continuous heterogeneity problem. Just like 3D variability is a different kind of model for modeling that. Of course, like I said, 3D variability is a first step in this direction, and the linear subspace model is a simple one, but it turns out that in practice it can capture much more than even more complicated models that have been attempted. But it is very different from multi-body refinement. What you will tend to find if you do run 3D variability analysis and multi-body refinement on the same data is that 3D variability analysis will show you, without the user having to say anything about what kind of flexibility or motion there might be, it will directly show you how the protein moves. From that information, you can manually create masks around regions that you think are rigid based on the 3D variability results, which were totally unbiased and only data driven. And then those masks will help you get higher resolution structures of parts that are in fact somewhat rigid. Of course, in real proteins, motion is never fully rigid. Like no part of the protein, if the protein is bending, it's not bending around a perfect hinge joint. And so the notion that we're dealing with rigid objects is false. But it's still a worthwhile approximation. But in order to figure out which parts of the protein move together and which part moves separately, 3D variability analysis gives you all that information without knowing that ahead of time.
Okay. Last question, I think, before we can move on to examining the results of our experiment. how does the 3D variability algorithm or analysis compare with CryoDragon?
That's a great question. So CryoDragon is a recent attempt also in this direction that is using a very similar notion to 3D variability analysis, but a different model where this linear subspace is actually modeled with a much more complicated deep generative model from a type of model that has been explored widely in deep learning literature. It's a very interesting idea in terms of a sort of experimental or yeah, it's an interesting idea in terms of something that could lead to something useful. At the end of the day, the model that's used here gives two things. One is expressiveness. So a more complicated model can potentially capture more kinds of heterogeneity. On the other hand, the model also determines whether or not it's possible to actually computationally infer the parameters of that model. So in 3D variability analysis, what we're able to show is that because the model is simple enough and we've come up with this algorithm that works for this model very well, we can actually resolve high resolution detailed motion of individual alpha helices moving by a few angstroms, even large subunits of big complexes moving in a very stable way that doesn't require any tuning or any changing of parameters or any sort of hand-holding. And it resolves fundamentally more information about motion and about heterogeneity than, for example, a much more complicated model where this part is replaced with a deep generative structure, as in CryoDragon, which can only resolve very large coarse motion of big things like ribosomes and spliceosomes. It's hopeful that as these models all evolve and as everyone continues working in this space, we'll be able to come up with models that are better and better at actually modeling motion while still allowing us to infer what that motion is and structures at high resolution. So I'd say in terms of practice, it's definitely worthwhile for everyone to try all the methods available. 3D variability analysis definitely works out of the box in a lot of cases where I don't believe other methods are able to operate, regimes where other methods are not able to provide results at all.
Thanks.
Great. Okay, so the first result I'm going to show is a structure and variability analysis of a Plasmodium falciparum ADS ribosome structure. And this is a protein, this is a ribosome that has been studied. It's a benchmark data set that many people use. And usually it's refined under a homogeneous refinement where we assume the entire structure is rigid. With 3D variability analysis, by starting with a consensus refinement of the ribosome, we're able to find actually multiple, in this case, 4 different kinds of conformational heterogeneity. This figure that I'm showing is actually directly from the paper, and I suggest anyone who's really interested in this data set to look at that. First of all, what I want to show is that once we run 3D variability analysis, what we find at the end of it is that we get multiple variability components. I'm showing the first 2 here in a 2D scatter plot. Each dot represents one particle image. So we're actually now, with this method, able to get sort of quantitatively or qualitatively an image that looks kind of like that conceptual diagram that I had before, where we're thinking of the space of particle images, and now we can see the distribution of particles in that space, forming these 2 kind of largish clusters, and one of the clusters has lots of spread in it. Variability component number 1, it turns out, represents a type of heterogeneity where part of the head subunit of the ribosome goes missing. And variability component 2 is a different kind of rotation that we're going to see. What I'm actually going to show you next, after we've looked at this figure, is what the variability components actually look like in terms of motion. And the easiest way to resolve that is in video form. So this is the first variability component. And what we're seeing in this video is a change in the electron density of the basic ribosome structure where part of the head region is appearing and disappearing. Just play that again. This is the second component now. This time we're looking at a type of motion where the small subunit of the ribosome is rotating, ratcheting. And I'll play this one again too. What's important to recall here is that all of these different kinds of motion are being solved at the same time by 3D variability analysis from the same set of particles. What we've done is we've asked for k equals 4 variability components. And so as we solve more and more components, each one resolves a different kind of motion that might be present. I'll just play this one again so you can see that ratcheting motion. In order to generate these movies, what we've done is take the basic electron density structure and add, let's say, our variability components. If I go up a couple of steps, along variability component 1, we have values that range from -400 to +400, let's say. So what we've done is we take the basic electron density structure and add -400 times the first variability component, and then generate volumes along this dimension all the way to +400. That gives us a sampling of volumes in the family that we're describing with 3D variability analysis. Now the third component, if we look very closely, is a different kind of motion of a small subunit. The small subunit is moving left and right. I'll play that again. This is a smaller motion now, a smaller kind of change. But you can see that the small subunit is overall shifting left and right. And the fourth subunit is rotation of the head part of the small subunit around this transverse axis. So these 4 different motions are all present at the same time, and the overall molecule has many different kinds of motion ability. Okay, the next example we're going to look at is a data set that we also already saw earlier, the NAV 1.7 channel. And in that case, we're able, this is now, like the ribosome is an example where many people who've attempted to work in the space of conformational change, including some of the most recent techniques that have been developed, ribosomes are often a great choice for an example data set because they're very large, lots and lots of signal and a few different kinds of motion, and very easy to work with. In real life, of course, smaller proteins and membrane proteins and all kinds of others give many challenges, but 3D variability is, like I said, the only method, as far as we know, that is able to resolve molecular motion or electron density motion for these kinds of proteins. So in the case of the ion channel NAV 1.7, sorry, I'll just go back. The first component, which again we saw earlier today, is a component that shows flexibility of the 2 fabs that are bound to the structure, as well as bending of the overall membrane domain of the protein. The next component, which is, we actually solved 6 components, and I skipped a bunch of them just because these ones were more interesting to look at in video, is one where we're seeing the motion of both of the other subunits that are not attached to the fabs. And we're seeing that there's a huge amount of motion in these transmembrane helices on the peripheral ends of the protein. And that's actually what limits their resolution when we're doing a refinement. Now, this is exactly the kind of result that on a larger protein would suggest to us that we should actually start trying to do local refinement by creating a mask that includes only part of the structure that's moving. The trouble with that, of course, is that local refinements and multi-body refinements that we can talk about can only work when the amount of electron density, the amount of matter inside each of the masks is very large, like 150 kilodaltons or more. So a fragment of a protein so tiny, we can't potentially independently align with other techniques. 3D variability is the first method that can show us the motion of these very small detailed parts of proteins. These are 3 components from the NAV 1.7 channel. We've seen the first and the last one already. The middle component actually shows shifting of the 4 helix bundle from one side to the other. Got this already. The next protein we're going to look at is another one that we've already looked at today as part of our practical study. And this is a GPCR complex, the Cannabinoid Receptor. And the first point I want to make here is that with membrane proteins, there's an important detail about 3D variability analysis that's very important to keep in mind, which is that the micelle itself generally presents a huge amount of variability. And that variability is stronger than the variability of the protein. So if we use 3D variability naively on the entire data set with all the particles and nothing else changed, what we end up with are results that are shown here in part B of the figure. So if you look at these 2 images, they're from one side and the other side of variability component 1, solved just directly. And this circle is in the same place in both cases. And what you see is that it's actually, all the variability is actually in the shape of the micelle changing along that axis of the family, and same with variability component 2. And they change the micelles in different directions. So this, in this way, 3D variability shows us that the micelle itself has a huge amount of variability, which is great to know, but not what we're most interested in. What we're most interested in is the flexibility and variability of the protein itself. Luckily, with 3D variability analysis, we are able to provide it a mask so that we can control that it only looks for variability inside of some region of voxels. Right. So using this mask is very important, and it's what allows us, along with 3D variability, to solve for continuous flexibility of interior parts of this membrane protein. So the next videos I'm going to show are what happens when we do that. And once again, this is a striking result for 2 reasons. One, because we're looking at the experimental motion dynamics of this molecule. This is not simulated by molecular dynamics or some other kind of model or normal modes or anything like that. This is actually what's happening in the electron microscope images. But the parts of the protein that are moving are very small. So this transmembrane domain now is less than 50 kilodaltons, less than even 40 kilodaltons, I think. And so we're seeing its motion right now in one direction, where it's moving toward and away from the rest of the complex. In component 2, we see a different kind of motion where the transmembrane domain is now moving in the opposite direction relative to the main part of the structure, and this 2-helix bundle at the bottom is also moving closer and away from the rest of the structure. So that's an interesting kind of motion. And this is exactly what I meant when I was saying that rigid body models, for example, multi-body refinement, can't capture this kind of motion because no part of this protein is moving in a rigid sense. And finally, component 3 shows us even more interestingly full twisting of the transmembrane domain of the 3D variable of the GPCR complex. So this time we're able to see motion again of just a very small part of the protein, but we didn't actually tell the algorithm that we want to see motion in the transmembrane domain or any part of it. We just asked for the first 3 most important types of variability in the data. So this is just showing all 3 of those again together. Okay, the last example I'm going to show before we get back to our own practical processing that we were doing is a ribosome assembly. This time is an example of not continuous flexibility, but discrete heterogeneity that 3D variability analysis can also be very useful in detecting, beyond, including and beyond what things like heterogeneous refinement and Ab-initio reconstruction can do. So for this data set, I'm actually just going to show you, what the protein looks like. I have it open here in Chimera. This is a consensus refinement of all the particles in this ribosome assembly. This is a large ribosomal subunit, and in the data set of several 100,000 particles, it comes in many, many forms with different subunits attached or detached. Sorry, what's my mouse? So when we're looking at this ribosome assembly, we can see that as we change this threshold slider, we can see parts of the protein appear at very low density, meaning that they were present in very low occupancy. And as we increase the slider, we can see other parts of the protein disappear and appear. So it's clear just from this that there's some substantial amount of heterogeneity in this data set. Now with heterogeneous refinement and Ab-initio reconstruction and so on, we can sort out many of the conformations, and we can also do sub-classification. Every time we find a different conformation, we can do heterogeneous refinement within those. And it's a similar strategy to that the original authors used a few years ago when they processed this data to find some 15 or 20 different conformational states, including many finely distinct substates. We can actually do something interesting with 3D variable analysis on the same kind of data set. And what that looks like is putting all those images into 3D variability analysis. And instead of the variability components now encoding for motion, because the variability in the data set is to do with appearance and disappearance of subunits, that is actually equally well captured in the linear subspace model. And so what we can see there is, actually I'll just switch back to the results, if we think of the space of variability components, in this case, we solved it with 4 variability components, we can see that in that space, the particles form these very clear clusters. And this is almost exactly like that diagrammatic figure that we had before, conceptually thinking of how particles are arranged in the space of particle images. With 3D variability, we can actually look at that in a few dimensions and see how the particles are different from each other and how they cluster. Again, this is without actually doing any classification prior to this. We just did one homogeneous refinement of the entire data set and then fed it to 3D variability analysis. So we can take those particle points and then actually do clustering via any clustering method that you like. In CryoSPARC, we've implemented a simple Gaussian mixture model clustering in this low dimensional variability space. And we can separate this into 8 different clusters. And then I'm showing at the bottom here a few other different ways of doing clustering with some more advanced machine learning techniques. But even the simple clustering can give us 8 different conformational states. We can take the particles, separate them literally by their variability component values, like take all the particles in the gray cluster, all the particles in the green cluster, and reconstruct them and end up with 8 different reconstructions, 7 plus 1 special case, all of different conformational states of that ribosome assembly. There are states that have only, I don't know what the different names of the subunits are, but each different subunit, whether it's the sort of head or foot region that disappears or the 2 arms on either side, they come and go in different configurations of the molecule, which we can clearly separate as different clusters in our variability space. Just making that more concrete in practice, we can also actually inspect that distribution, and we'll show how to do this in the practical as we go. You can inspect that distribution of particle points in the 3D variability components right inside of CryoSPARC to be able to see those different clusters and their positions in the 3D variability space. Okay, so that's another use case for which 3D variability can be useful. The next thing we're going to do is go back to our practical session and actually start looking at the 3D variability results, which I believe for most people may be complete or may not be complete by now. I'm not sure how long we've been talking since we started them, but I'll show how to interpret those results and various other detailed parameters and so on of 3D variability analysis now that we're talking about it. So just digging into this job a little bit, 3D variability analysis starts with a mask, which in this case, we provided a mask that includes the entire protein. We didn't want to ignore any part of it. But for the GPCR structure, for example, as I mentioned, and for all the other membrane proteins that we show results on, you do have to exclude the micelle in order to be able to see the variability of the protein itself. Once we do that, 3D variability analysis loads up all the particles, creates the V0 consensus refinement structure, and that comes from the particle pose alignments that we already have as input. And then it starts by randomly initializing itself and starting to go through iterations of the optimization procedure that allows us to solve for the variability components. At the end, it results in the actual variability components, which we can see here as central slices. They're all 3D volumes of positive and negative values. We can see them as central slices with red and blue values encoding the positive and negative parts. And we also see the distributions over those latent particle coordinates for all the particles. Now, 3D variability, the job itself, is not meant to be something that you can directly get results out of in order to be able to display them or make those videos. So the next thing we're going to do with the output of 3D variability, particles and volumes, which includes the variability components, is use a 3D variability display job. And I'll just clone that job so everyone can see how to build it. What you'll need to do is go to the job builder, create a 3D variability display job, and to it, why did I just do that? Go to the job builder, create a 3D variability display job, and to it connect the particles and volume outputs of your 3D variability analysis job. And then in the parameters for the display job, we have different modes for creating different kinds of outputs. Simple mode creates the movies that I showed for all of those different data sets. There's also cluster mode, which does the clustering that I showed for the ribosome dataset, the assembly of different ribosome states. And intermediate mode, I won't go into, but it allows you to reconstruct structures from different subsets of the particles. So we'll stay in simple mode right now and leave all the other parameters the same. We want to create 20 volumes along each of the dimensions. And the only other parameters we're going to change are to downsample the output. So in this case, you put in a parameter of 128 and crop the output also to a size of 100. The reason we're doing this is because this job is going to create a series of volumes, and the space required for those can be very large if you leave the box size really big. But we know that we're not looking at this structure at better than 8 angstroms-ish, since that's the parameter that we applied before. So we don't need the full resolution of the data, nor do we need to keep all the extra white space around the protein. So if we crop the protein, crop the box down to a size of 100, we'll save a lot of space in our final results. So you can go ahead and queue that job. Now I've already done that, so I have this 3D variability display job present here. When you open up the 3D variability display job, it again reports the images of the consensus structure and the 3 variability components that were solved, and an interactive plot that allows you to browse through the actual Z values, those latent coordinates, to see if there are any patterns or anything present that you might want to investigate. In this case, we can see there isn't really any clustering, and maybe what we're looking at is actually just continuous flexibility, and we'll find out exactly what we're looking at in a minute. The rest of the job is just creating outputs, and those outputs are in the form of what we call volume series. There are 3 of them. So the first one we can just download, volume series. And that should take a few seconds. That's going to be downloaded as a zip file of all the volumes that we that make up the series that show us that movie. And I'll show you exactly how to visualize that right now in UCSF Chimera. So I unzipped that directory, and inside that directory, I'll just show you, are a series of frame volumes. Each one is one position along the flexible motion that we wanted to resolve. So frame 0 until frame 19. Now to visualize those, what we're going to do is open up UCSF Chimera, our favorite tool for this, close all the other volumes that we had, and then choose the volume series option. By the way, everything I'm showing you here is also present in our tutorials online, which I'll show you in just a minute. In that volume series tool, we're going to open up our volume series. So I'm just going to do that. The open column, just want to verify the name, project 34, job 60. So I will find project 34, J60, and select all of the MRC files by holding Shift there. That opens up our volume series. So now I'm going to adjust the threshold at which we're looking at this volume. And then in the volume series tab, I can start playing the movie by hitting play. I also want to oscillate the movie. I'm going to set a maximum playback speed of 20 frames per second and cache the renderings just to make it a little cleaner. And then maybe 20 frames per second is, yeah, okay, that's good. So what I'm seeing now in this first variability component is an interesting kind of flexibility in our molecule. You can see the peripheral stalks, both of them, moving closer and further away from the body of the protein. Now, I should have said when we started here that the set of particles that we're using is actually a subset of the whole data set, and it contains only the particles that went into one of the 3 rotary states. So I already used heterogeneous refinement to separate the 3 rotary states, and then I took the particles from just one of them, and we did a 3D variability analysis, and that's what all the practical students should be doing right now. And so our first variability component is this interesting motion of the peripheral arms away from and closer to the body of the structure. We can see that from different directions. We can also change the thresholds and see it in more or less detail as you wish. All the instructions for how to do this visualization are all available. First of all, if you go to 3D variability analysis, 3dva.cryospark.com, you will come to our 3D variability analysis landing page. For anyone who's interested in reading it, you can read the preprint, and you can also go to our tutorial series, which are online. There's two parts. The first part explains how to run 3D variability analysis. The other one explains more advanced concepts, masking, clustering, everything that I've talked about, and how to make these movies. So all those tutorials are all online, so I won't go into too much more detail about that. And this page also has the videos and everything else from the paper. Right. There is one other result that I want to show. And for the VATPAs, we can start looking at the other variability components by downloading those. So I will just do that. For my display job, I'll download series 1 now. And that I'll just get here in a moment. In the meantime, I will show you a video from a result of 3D variability analysis running on a larger data set of 20S proteasome particles. The same data set, the same particle, and in fact the same data set, but the entire set of images that we used yesterday. Actually, I'll come back to that since now my variability analysis is finished downloading. So what I can do is close this first volume series. Oops. Close series, and then open the next series, which is component 1. And this time we should see a different kind of motion present. So adjusting the thresholds to a reasonable level. And play. Just look that up. And what we see here now is instead of the peripheral arms moving towards and away from the central stalk, we see them bending in a sort of shearing direction. So this is another kind of flexible motion that our molecule undergoes.
Quick question here. Is there a way to actually record that movie, or rather series of frames, in Chimera?
Yes, actually we cover all of that in our tutorial. So if you go to the first part of the tutorial, there's a section on exactly how to generate these movies at the bottom, visualization that does all the things that I just said, and has commands for recording a movie of them in Chimera.
Perfect, thanks.
No problem. Okay, so that is, we're going to stop there with this data set. But I do want to show one last result from 3D variability analysis on a symmetric protein. And this is a very interesting example. It's something that we've been talking about a few times. When you have a symmetric molecule, how can you detect things that break that symmetry if you use the symmetry during refinement? So this is an example of running 3D variability analysis on 20S proteasome particles that when we did refinement, we used all the particles and we enforced the symmetry, and therefore our consensus refinement is symmetric. But what's really interesting is that with 3D variability analysis, we can actually resolve symmetry breaking flexible conformational changes amongst these particles. And so to do that, actually, I think I have the video loaded here already. Yeah, to do that, I will just play this video. 1 second. What we see in this video are 5 different conformational variability components for the 20S proteasome. And we're going to see them all in detail in a second, so I won't stop here. Sorry, I'll just let the movie play a second. The first component shows us extension of the barrel. So this is a symmetric type of motion where all the subunits are doing the same thing, and the whole barrel is kind of extending a little bit. The second mode shows us rotation of the end caps of the barrel. And again, this is a symmetric motion where all the subunits are doing the same thing. Although we didn't ask 3D variability for a symmetric solution, I just found these as the most important kinds of variability. Now the third mode, or the third video here, actually component 5, is showing us an asymmetric bending. And I hope everyone was able to actually see that. What happens here is that the protein, sorry, what was in front of that? The protein bends across its axis. And that's not a symmetric motion. Now, different subunits are doing different things in this kind of motion. Some subunits are coming closer together and some are moving further apart at the same time. So that's an interesting result because it means that we can start to resolve symmetry breaking conformational changes even with particles that we've assumed so far to be symmetric. Now, in order to get this kind of result, you need to use symmetry expansion, which is another job type in CryoSPARC. I won't get into the details of how to do that. I believe that's in our tutorials as well. So with that modification and using all the particles from every different symmetry related position, 3D variability analysis can resolve these kinds of flexibilities. And I believe that is everything we wanted to talk about on this topic. So we can jump over to any questions at this point.
Great. Our next set of questions are all about how to actually use 3D variability. First of all, is there a way to use 3D variability on a specific region of the protein instead of the entire structure?
Yes, and that is definitely what we did today or what we explained by using a mask. And I'll just come back to those slides. So with the GPCR example, for example, we only wanted to look at conformational changes in the protein region, not the micelle. So we created a mask that only excludes everything except for the protein and use that mask in 3D variability analysis. In order to actually do that, you can simply, when you're creating the 3D variability analysis job, you will see that one of its inputs is a mask slot. And you can connect any mask to that, including one that you've generated outside and imported into CryoSPARC.
About masks specifically, what actually makes a volume a mask?
Nothing in particular. I mean, masks are also stored in MRC files on disk. The main thing is that a mask only contains values between 0 and 1 typically, and is kind of treated differently when we're actually doing processing. You wouldn't use an electron density as a mask or vice versa. And so CryoSPARC thinks of them as different types of objects. Under the hood, they're actually the same. When you're importing MRC files, volumes, you need to tell the import job whether you're importing a mask or a volume.
You had mentioned that in order to be able to resolve motion of a certain amount, you actually need to have all of your data at a certain resolution to be able to see that motion. So can you please explain why 8 angstroms was chosen in this case, or rather in our specific tutorial case as the filter resolution parameter?
Great question. So to clarify again, this filter resolution parameter is a very important parameter that should be set for 3D variability analysis. And in fact, you have to set it to something to run the job. And what it does is that it enforces that the 3D variability components that we solve are limited in their resolution. And what that effectively means is equivalent to saying that we're looking only at the structure up to 8 angstroms or around 8 angstroms. It's not a sharp cutoff, it's kind of a smooth fall off. So we're looking at the structure in the region of around 8 angstroms. So we're able to detect motions around that magnitude, maybe up to 10, 15 angstroms is fine of motion. If we knew that we can already resolve the structure down to 3 angstroms, like with the GPCR, then we would set this filter resolution to a much higher resolution, like 4 angstroms. So we know that we are able to look at higher resolution details and therefore resolve smaller, more detailed motions. So this is a filter parameter that you should really experiment with, and you can run 3D variability analysis multiple times with different filter resolutions. They should always be below the overall resolution to which you've resolved the structure so far, since you can't really trust information beyond that anyway. And different resolution limits will resolve different scales of motion, typically.
When it comes to our consensus refinement, is there a resolution at which it makes more sense to perform 3D variability analysis?
Sorry, can you say that again?
Is there a resolution at which it makes more sense to run or try 3D variability analysis?
Not really. The nice thing about 3D variability analysis is that it's kind of agnostic to the resolution. I mean, other than the fact that the motion you can resolve is of different sizes when you look at objects at lower resolutions. Other than that, the method is overall the same. And heterogeneity in a particle can occur in so many different ways. Even if you have a 5 or 8 or 10 angstrom reconstruction, you could still find conformational heterogeneity. And in fact, that may explain to you what is the reason that your resolution is limited, is because you have a huge degree of conformational variability. So I'd say you should try it at any resolution, but if you can already resolve your structure to a higher resolution, that tells you that the amount of flexibility in the structure must be less than what would ruin a structure of that resolution. So if you can see the structure at 4 angstroms, you know that overall the structure is kind of rigid up until that point. There may still be motion finer than that, which is exactly what 3D variability analysis can show you. But around that resolution, you probably have a relatively conserved structure.
Okay. And in terms of actually assessing the quality of the result, is it possible that we can be seeing or introducing artifacts into 3D variability? And if so, how can we identify them? And more generally, what are the approaches for actually performing validation of a 3D variability analysis output?
That's a great question. Some of these things are still evolving topics, and I think we don't have, for example, something like Fourier shell correlation for 3D variability results. It's in principle maybe possible to do something like that, but the most important, so some of the mathematical properties of the 3D variability analysis algorithm tell us that there aren't too many ways for the algorithm to give an incorrect result if there actually is variability present in the data. So right now, the answer is if you see a variability result that can clearly be visually explained as either motion or association or dissociation of subunits or other kinds of heterogeneity that are plausible to occur, then that's a very strong signal that that's what's happening in the sample. If you don't see something like that, it may mean that either there isn't enough signal or enough particles to resolve any real heterogeneity or variability at all, or that pretty much that's what the answer will be. So in those cases, you shouldn't trust the result, mostly because the result won't have anything meaningful or interesting to draw from it insightfully anyway. But if you do have that kind of result, you can trust it thanks to the mathematical properties of this algorithm. First of all, that it doesn't depend on initialization. So you can't get an incorrect result just because the algorithm was initialized incorrectly, in contrast to, for example, 3D classification or even refinement and so on. Secondly, it can't miss anything that actually is present because it will always solve these variability components in order of importance. So the first one it solves is always guaranteed to be mathematically the largest source of variability in the particle images. And if that turns out to be something that is unintelligible, then you can be sure that none of the other variability components that could have come after that you may not have even solved are anything very useful. But if you do see something important, then the properties of the algorithm, and this is something, of course, that may be contentious depending on who's asking, the properties of the algorithm do provide those guarantees that we're looking at something that's actually in the data.
Thanks. Is the 3D variability job very memory consuming, and approximately how long would it take to run on a standard sized data set?
So it's actually surprisingly fast. It probably takes about the same amount of time, maybe twice as long as doing, for example, a heterogeneous refinement or something like that. Memory intensiveness wise, it's usually not, because we don't usually use it at the full resolution of the images, depending on how fine of motion you're looking for. And it's totally plausible to be able to resolve 3 or 5 or 10, I think I've done even 20 variability components. It's really limited by the GPU memory, but that doesn't seem to be a big bottleneck right now, memory usage.
Perfect. And you kind of touched on this a little bit before, but in terms of strategy, what's the best approach to utilize 3D variability in a sample that has both conformational variation and compositional variation?
Great question. So the optimal strategy, I would say, is to do two things in parallel. One, take all of your particles without trying to classify them at all, put them into 3D variability analysis and see what you get. That track will show you an unbiased, unmodified view of what kind of heterogeneity there is in the sample, whether it's discrete or continuous or any kind. If there is continuous heterogeneity, and if there's both kinds of heterogeneity, what I'm imagining in this scenario is that you have a particle that exists in multiple discrete conformational states, and each one of those states also has within it continuous flexibility. And that is very common, since that's probably how most particles work. In that scenario, the first time you run 3D variability analysis with all the particles, you're likely to only resolve the fact that there are multiple conformational states. Once you've done that and you can see that that's the case, it's a good idea to use heterogeneous refinement or the clustering version of 3D variability display, which you can read the tutorial for, just like I showed with the ribosome data, to separate the particles into subclasses, each one representing one more or less discrete cluster in the data. Now, within that cluster, there may still be continuous flexibility, and you should apply 3D variability again to that subcluster of data and see what kind of continuous heterogeneity you find. That's probably the best strategy in almost all cases. Try to resolve all the discrete heterogeneity you can, and then apply 3D variability to each subclass separately to get at all the flexible motion. On the other side, use 3D variability analysis with all the particles to verify that you found all the discrete clusters in the first place.
Great. And today, our examples for 3D variability didn't include very small proteins or protein components. Can we use 3D variability on very small protein components like in the 40 kilodalton range?
So if the overall protein is 40 kilodaltons, it's amazing that you have got a structure at all that you could use 3D variability analysis for. If we're thinking of a small protein, maybe in the 150 kilodalton range, that has a small subunit that is 40 kilodaltons or so, that is what the GPCR example that we looked at today was about. This transmembrane domain is only about 40 or 50 kilodaltons. And we can see all kinds of continuous, non-rigid flexible motion in that small subunit. So definitely, and in fact, we can even see motion of this GI tail of the G protein. I'm not sure of all the names of the components of a G protein complex, but this little 2-helix bundle, we actually see that moving as well in one of the variability components. I'll just find that one. This one. In this component, we actually see, no, sorry, wrong one. In this component, we actually see that tiny part of the protein moving. You can see this 2-helix bundle moving closer and further. So even tiny pieces of that small size, their motion can be resolved. So I would say try it and find out what happens.
Perfect. And last question, can you clarify if we need to impose symmetry in 3D variability?
You can't actually enforce symmetry in 3D variability. So if we look at the 3D variability job, it does have a symmetry input. But if you put anything here other than C1, the job will fail and give you an error message saying you shouldn't use symmetry in 3D variability analysis and to use symmetry expansion instead. The idea with symmetry is that you should take your refined particles and break the symmetry by doing symmetry expansion, which creates copies of the particles for each symmetry-related rotation around the structure. And then each of the particle images treated as independent in each of those positions. After symmetry expansion, 3D variability can then resolve both symmetric and symmetry breaking versions of flexibility. What you can also do in that scenario is create a mask. If you're only interested in symmetric flexibility, create a mask that only includes one subunit of your symmetric molecule. Then you'll be forcing 3D variability to only find motions of that subunit that all the subunits have within them, but not coordinated motion of the entire molecule across the symmetry, if that makes sense.
Thank you. Those are all the questions we have on 3D variability.
What, I do have one question also about just regarding that fact on cooperation and coordination between these. Should we think of each of these components of variability as being uncoupled and discrete from each other? Because if they were coupled, then they would be a different kind of flexibility that would show up as the combination of all those. Are they really kind of to be thought of as uncoupled and uncorrelated?
That's a great question. And the answer is definitely no. We can't in general. I'm just going to switch to a slide where I have an image that might help with this. We can't in general assume that just because two variability components were found and they clearly capture different motion, that those motions are not physically connected. The reason is that the variability components are only encompassing or capturing essentially what are eigenvectors or subspace directions in the linear subspace model. But it's perfectly possible for a particle image to have some amount of each of the variability components. And so the motion that you see in each variability component, you can't say that it definitely happens by itself and the other motion in the second variability component happens by itself. It could very much be true that many particles have both kinds of motion together and many particles have the opposite motion together, or they're somehow one way or another correlated, or rather dependent on each other. We can say that the two types of motion both happen, and the two types of motion are orthogonal in the sense that they're describing different kinds of change in the density. But across the particle data, and when we look at the distribution of particles across the variability conformation space, this scatter plot will often, just like this one, have a structure that shows us that these variability components are not independent. They may be orthogonal and they may explain different motions or different changes, but particles, it may be true that if a particle is on one end of one of the variability components, it's more likely to be on one end or the other of the second variability component, and therefore they're not independent. Hopefully that is making sense. It's if someone is looking it up, it's a difference between independence in a probabilistic or statistical sense versus uncorrelated. Uncorrelated just means that you've taken out the linear correlation between two things. Independent means that knowing one of the things doesn't tell you anything about the second thing. So we can, in 3D variability analysis, we know that the variability components are uncorrelated, but they're not independent necessarily.
Thank you.
Great. Any other questions or anything to talk about? I think that's actually the end of all the practical and slides that we wanted to show. I have one slide left about local and focused refinement, just to indicate that CryoSPARC has these capabilities. If once you do 3D variability analysis, you find out that, okay, it really looks like I have multiple parts of my protein that are rigidly moving, then you can separate your structure into multiple components, rigid parts, and do local refinements of each one. We also have a tutorial online that goes through this in a lot of detail, so I won't cover it any further. And that's the end.
Awesome. We did have a few questions that have been kind of hanging that I wanted to address if we had time.
Sure.
Taking us back a little bit back to the beginning. When we're looking at particle picking in a crowded micrograph scenario, and then we did the example with the AAA plus Unfoldase where we actually did windowing to make sure that we can actually see individual particles in each of our classes. Those are just masked. Don't we need to re-pick from the data set with a smaller box size, or rather re-extract with a smaller box size? That's
a good question. The answer is no, we don't need to. Let me see if I can find that again. Let's see, Unfoldase. So when we did 2D classification without the windowing, what we found was that we got lots of classes that have multiple particles in them. What that means is that our original particle images, which we can see in this import job, have many particles in the box. That's actually not a problem. Even though it seems like we would want one box to only contain one particle, it actually doesn't hurt us to have particles in the periphery. They are present in the images, but when we do 3D reconstructions, the signal anywhere in the image that is far enough away, I mean, it's very tight crowding, like in this example, theoretically does have an impact. Not that there's anything we can do about it because the sample is already frozen. But the particles that are far away, they contribute in the 3D structure to density that's outside of the main internal first particle. And all of that density gets masked out during a refinement. And so we don't actually care that there are other particles floating around. They won't ruin the structure of the central particle that we are solving as long as we're able to align that central particle repeatedly across all the images. So we don't need to re-extract. Once we did 2D classification with the windowing, we can be sure that we are able to align the central particle across lots of images, and that's what allows us to do reconstructions very well.
Great. Taking it back one step further to CTF estimation. If we can pull up a plot again of the CTF, specifically looking at the tone rings, we'd like a reminder on what exactly the right half and the left half of the image correspond to.
Sure thing. Sure thing. Let me just find a good example. Okay, this is a good one. So first of all, when we're looking at these tone rings, we have to recall that we have the real space image, which is the actual image captured in the microscope. That's this image where we can see the particles. And we have its corresponding Fourier transform. Now the Fourier transform, the mathematical operation of taking a Fourier transform, converts our image that was in real space into a different representation. It still captures all the same information content, but it's a totally different representation where each pixel here in this image, each dot, corresponds to one spatial frequency component in the real space image. What we're looking at in this picture of the Fourier transform is, on the right-hand side, the experimental Fourier transform of this image. Now, normally if you study Fourier transforms or something, you will see that when you take a Fourier transform of a 2D image, you get a square 2D Fourier transform that has circles that go all the way around. That is true, but a Fourier transform of a real space object is actually redundant in the sense that its left and right half, or top and bottom half, or whichever way you cut it in half, they're identical, just mirror symmetric. So we don't need to see the whole thing, we only need to see half of it. And so the right half is exactly the Fourier transform of this raw image. What we should be seeing in that Fourier transform are these rings, the tone rings. Those are the experimental tone rings that show us where there is signal coming from the sample. And this white band is, like we kept discussing, the ice signal. On the left side now is, in Fourier space, the representation of the simulated contrast transfer function. If this was a perfect image with no noise and no ice background and a perfect sample, we would actually see what we see on the left side in the experimental image. Perfect tone rings that go infinitely far out within some reason. In real life, we don't see the tone rings go that far out. But the simulated CTF, which doesn't have any experimental data in it, shows us what those tone rings could look like if they did go very far out. And what we want to see here is just that between the experimental and simulated data, the rings all match up. That's just a sanity check that our CTF estimation is right on. And we, in our simulated CTF, we've just about matched the tone rings that we can see in the example, the actual experimental data.
Great, thank you.
Okay, I think we probably have exhausted all the energy for today, and I suggest we would adjourn for today's meeting. And I'd like to share the announcements for tomorrow, and we would have 2 speakers in the morning. And can you see my slide?
Yes, we can.
Okay, so we will start at 9 o'clock as scheduled, and through the consensus of all the instructors, they are happy to have an over an hour and a half general discussion with the entire participants. So we have a lunch break at 11:45, and we will reconvene at 12:45. And so for those who still have questions in the last couple of days tutorial, you're free to raise the question again through Q&A box. And Sarah is kind enough to lead the discussion tomorrow. And then after that, we will officially adjourn for the general audience. And then we would have a small group of the practical participants, and some of them will present their own experience, and we'll go in depth in some of the steps that we have gone through. And so that's what I want to encourage you to reschedule your time for tomorrow. I think in my experience in the last couple of days, the question and answers period is outstanding. I think you can probably learn a lot through raising the questions or just listening to the question and the answers. And again, I like to thank Ari and Sarah and Stefan for their outstanding presentations. And this has been a wonderful day. Thank you very much. And have a good evening, and I will see you tomorrow.
Definitely.
Thank you.
And thank you all for listening and paying us attention for so long.
And organizing.
This has been a wonderful day. Thank you.
Thank you.
Okay, good. See you.